Welcome to the Philippine Science Heritage Center's new Salinlahi Science Center, honoring our top Filipino scientists and their game-changing contributions to the world of science. Know more about all of them and let the Salinlahi Science Center be your gateway into their minds. Salinlahi.
Are you ready for the new Transactions Nashville? The Transactions Nashville ISS N01158848 has been published by the National Academy of Science and Technology Philippines since 1979. Traditionally, published papers presented during the Nashville's annual scientific meeting to promote science-based policy discussions of and recommendations on timely and relevant national issues as part of its functions as a National Science Academy. Starting with Volume 43, this journal is now open to contributions from the global scientific community in all fields and will be subjected to double-blind peer review. Accepted papers will be published in the Transactions Nashville website. And here is a quick preview of the website. The papers for the journal include conference proceedings such as messages, plenary papers, resolutions of ASM, etc., perspectives, articles including review and feature articles, biographical or autobiographical essays of NAS members and other outstanding Filipino scientists. The papers will be uploaded as soon as they are approved for publication. Take note that all papers are downloadable. You can also zoom in or out depending on your liking. The backlog starting 2016 will also be published soon. The Transactions Nashville will be a key platform for the timely release of advisory documents of Nashville as part of its advisory functions. Let's make a timely and relevant Transactions Nashville. Collaborate with us by visiting transactions.nas.ph or email us at publications@nas.usc.gov.ph.
Before we begin, please be reminded of our house rules. Proper decorum is expected among all participants. Any act of disrespect and rudeness shall not be tolerated throughout the duration of the webinar. Kindly key in your questions for the panelists in the Q&A. These will be answered during the open forum.
Hello. Hello, magandang umaga sa lahat. And on behalf of the National Academy of Science and Technology Philippines and the Microbiology Consortium of the Philippines, I am pleased to welcome all of you to the second of the three-part series of the focus group discussions on microbes and climate change, focusing on terrestrial and marine environments. So I'm Aleta Iniguez. So I'm a professor at the Marine Science Institute, University of the Philippines in Diliman, and I'll be the moderator for today's FGD. So please do let your friends, your colleagues know that you are attending this activity. We have official hashtags, as you can see here, hashtag PhilippineNast and hashtag microbes and climate change. So please do use them so we can easily track and see the topics that we're engaged in for today. So just a few reminders before we start. Kindly key in your questions in the Q&A box in Zoom. For those streaming in the NAST Philippines Facebook Live, you may type in your questions in the comment section there. And the speakers will be answering your questions during the open forum that's scheduled later in the morning. Also, to receive an e-certificate for attending this activity, participants are requested to fill out an evaluation form after the program. So the link to this evaluation form will be announced later. So at this point, let us hear from academician Eufemio T. Rasco Jr., the interim president of NAST Philippines and the chair of the NAST Agricultural Sciences Division. So to give us the welcome remarks, please give a hand to academician Rasco Jr.
Distinguished participants, ladies and gentlemen, my dear friends of science, technology, and innovation. Magandang umaga sa inyong lahat. On behalf of the National Academy of Science and Technology Philippines, I'm honored to welcome you all to the second of the three-part series of focus group discussions on microbes and climate change, focusing this time on terrestrial and marine environment. Academician Asuncion Raimundo, NAST focal person on microbiology education, together with Biological Sciences Division Chair Academician Wendell Rivera, Academician Virginia Cuevas, and corresponding member Francis de los Reyes III, initiated this series of focus group discussions on microbes and climate change. The first FGD was held last November 2022. Previously, the discussion involved the updates and overall perspective on microbes and climate change and the network models for climate change mitigation and adaptation. Microbial and data systems, quantification of microbial injustice in the Philippines, research needs, and investments in addressing climate change, and the slow adoption of technology and methodologies in the country were also discussed during the open forum. For this session, the impact of climate change on plant microbes interaction, the role of microbes in the conversion of waste, and marine environmental monitoring initiatives will be discussed. This FGD specifically aims to highlight the importance of microorganisms in climate change and to incorporate microbial sciences into the current and future climate change initiatives and remediation processes to lessen the impact of climate change on our lives. We thank our resource persons, Dr. John Leach, Dr. Noel Sabino, corresponding member Francis de los Reyes III, Dr. Deo Florence Onda, Dr. Charina Gracia Banaay, Dr. Aleta Iniegues, and Dr. Rico Ancol for accepting our invitation to share their time and expertise in this very important and timely activity. Our sincere gratitude also to the organizing committee, headed by Academician Virginia Cuevas, Academician Asuncion Raimundo, Academician Wendell Rivera, and corresponding member Francis de los Reyes III for spearheading this FGD. Let me extend the Academy's appreciation to all of you who have given the time to attend this activity. I look forward to the excellent presentations and fruitful discussions during the open forum. Thank you and a pleasant morning to everyone.
Thank you very much, Academician Rasco, for your welcome remarks. So now, to give us a brief overview of the flow of this FGD and have a better idea of our speakers, please let us hear from Academician Virginia Cuevas, who is the chair of the organizing committee and member of the Biological Sciences Division of NAST. Academician Cuevas, the floor is now yours.
Okay. Members of the Academy, national scientists, academicians, young scientist awardees, distinguished resources speakers, friends, ladies and gentlemen, members of the Microbial Consortium of the Philippines, good morning to us here in the Philippines, and good evening to our participants in US and elsewhere. As mentioned by Academician Rasco, this is a three-part series, and part one of the FGD was held last November 19, 2022 with Dr. James and TJ, University Distinguished Professor, Michigan
wala na naman akong volume.
as our lead speaker. Can we have my slides, please? Okay. Next slide, please. Next slide. Okay, I will be giving the overview of this FGD. Microbes play important role in climate change. They are major drivers of global geochemical cycling, specifically that of the elements carbon and nitrogen, which are important components of greenhouse gases. Microbes are also critical symbionts of global crops. Microorganisms are important producers and consumers of greenhouse gases. Carbon dioxide is the #1 culprit for global temperature increase. This gas is responsible for 2/3 of total heating influence of anthropogenic carbon dioxide emissions. So this gas emission is due mainly to the burning of fossil fuels by the transport industry. And the transport industry is believed to be responsible for about 24% of the total global carbon dioxide outputs. Other greenhouse gases, nitrous oxide and methane, are produced by the agriculture industry, and the production of these gases are mediated by microorganisms. Next slide, please. Okay, this slide will be the agroecosystem or agricultural ecosystem. Now, nitrous oxide is mainly released through soil disturbance during cultivation phase of the agricultural field. It is also released during the cycling of nitrogen fertilizers, urine, and dung. The global warming potential of nitrous oxide is 310 times that of carbon dioxide over a 100-year period. Carbon dioxide is mainly released through burning of fossil fuels, as I have already mentioned, plant decay, and insect and microbial activity in soil. It is also absorbed in the process of photosynthesis by plants and stored in the soil and in the biomass of trees in the forest. Methane is mainly released from cows and sheep following digestion of plant matter. The global warming potential of methane is approximately 25 times of that of carbon dioxide over a 100-year period. Next slide, please. Global temperature increase has impacts on all components of the ecosystem. Increased soil temperature affects soil microbial communities and activities, and this has important implications for food security and global carbon and nitrogen cycle. Our first speaker, Dr. John Felix, distinguished university professor of Colorado State University, will discuss climate change impacts on plant microbe interaction. Our second speaker, Dr. Noel G. Sabino, will discuss microbes and climate change, adaptation and mitigation measures in agriculture. He will discuss how microbial activities can help mitigate global temperature increase through some innovative agricultural practices that will make agriculture industry adaptive to climate change and make it sustainable. Next slide, please. It is predicted that about 6.5 billion people will live in cities by 2050. The urban poor, which we have millions here in the Philippines, are highly vulnerable to climate change effects, as they tend to live in more polluted, less secure, and high-risk environments that drive inequitable microbial exposure. Sewage and wastewater systems are especially susceptible to microbiological influence that influence, damage, and impacts of climate change. Sewage treatment relies on microbial metabolic activities to degrade waste, producing carbon dioxide, methane, and nitrous oxide, which all are greenhouse gases that contribute to climate change. Our third speaker, corresponding member Francis de los Reyes III, offers idea to address this issue as he discusses the topic waste to energy. Next slide, please. Climate change also alters marine carbon pools. We know very well that one of the storage pool of carbon would be the marine sediment. This is referred to as marine carbon pump, which utilizes microbial processes to sequester large amounts of carbon in the deep ocean, helping regulate atmospheric carbon dioxide levels. Understanding seasonality of microbial communities could lead to insights into the changes in environmental conditions that broadly affect global cycles such as carbon dioxide. Our fourth speaker, Dr. Deo Florence Onda, will present data on long-term monitoring and studies of harmful algal bloom, HAB, populations in Manila Bay and Bolinao. This historical data will allow for correlation of HAB occurrences with meteorological patterns and regional atmospheric oscillations, such as the El Nino. The in situ collected data will help understand ecological systems and provide insights on the effects of climate change in the marine environment. This information will be helpful in crafting policies and management strategies for our marine environment, a much needed initiative in the Philippines. After the last speaker, we'll have an open forum. Therefore, thank you. Let us listen and actively participate in the discussion. Good morning to all here in the Philippines and good evening to our participants in US and elsewhere. Thank you.
Thank you, Academician Cuevas. So thank you for setting the stage on showing us the significance of microbes in our lives and the environment, and also the insights that we will be gaining from our series of speakers. So to introduce our speakers, may I now call on Academician Asuncion K. Raimundo, a member of the Biological Sciences Division of NAST.
Good morning, everyone. I have the pleasure of introducing the first three speakers, who are all my friends, colleagues, and collaborators. The fourth speaker will be introduced before his presentation. Our first speaker is Dr. Jan Illich, University Distinguished Professor and Associate Dean for Research at the College of Agricultural Sciences, Colorado State University. I knew Dr. Illich since the 1990s due to our collaboration and our research on rice pathogens, and we became close friends. She is a Filipino at heart, as she used to frequent Erie, and when she is around, we traveled together to tourist spots in the Philippines. And I also frequented her lab for some years when she was still with Kansas State University. Dr. Illich is a plant pathologist, recognized for her work on the molecular basis of plant disease susceptibility and resistance, and how these responses are influenced by interactions within the phytobiome. She did not share her awards with NAST. These are numerous, but I know she has been bestowed the Award of Distinction from the American Phytopathological Society, the largest professional organization of plant pathologists in the world. For its 112 years of existence, Dr. Illich is one of the only 16 individuals who received this honor. She was also awarded the Agropolis Foundation Luis Malasis International Scientific Prize for Agriculture and Food. She is a member of the National Academy of Sciences. She has served as president of almost all organizations she joined: the American Phytopathological Society, the International Society for Plant Microbe Interactions, International Society of Plant Pathologists, and chair of Agriculture, Food, and Renewable Resources for the American Association for the Advancement of Science. She will discuss climate change impacts on plant microbes interactions. Our next speaker is a colleague of mine at the Microbiology Division of the Institute of Biological Sciences at UPLB. Dr. Noel Sabino is young by my standard, as he was still an undergraduate student when I was already a faculty member. He is the microbial ecologist in our division now and teaches this course. He got his MS degree in microbiology and his PhD in environmental science at UPLB. While his research deals with bacterial diversity in honeybees, lactic acid bacteria, and also work on bee pathology. He is a curator for bacteria, yeast, and molds at the UPLB Museum of Natural History. He received the prestigious Professor William L. Fernandez Award for Excellence in Teaching Microbiology, awarded by the Philippine Society for Microbiology. He was also certified as a specialist microbiologist by the Philippine Academy of Microbiology. He will discuss microbes and climate change, adaptation and mitigation measures in agriculture. The next speaker is a professor from North Carolina State University, Dr. Francis de los Reyes III. He is a fellow University of Illinois alumnus, and we had some research collaboration leading to some publications with our students. He is a corresponding member of the National Academy of Science and Technology Philippines, meaning that he's a scientist of Filipino descent working abroad and has distinguished himself in his field, so he was elected to the Academy. Dr. De Los Reyes is in Philippine term a suki, as he gives talk whenever he comes to the Philippines and also online. He is actually an engineer, having gotten his BS Agriculture Engineering from UPLB and his PhD in Environmental Engineering from University of Illinois. I love to say this about him. He engineered himself to become a microbiologist, a molecular microbial ecologist at that. He has pioneered the integrated use of molecular, microbial, and biotechnological tools with biological process engineering to improve analysis, design, and operation of engineered and natural waste treatment and waste to energy systems. He's also involved in water and sanitation projects for developing countries, including the Philippines. He is a TED Fellow, and his TED Talk Sanitation has been viewed over 800,000 times, maybe 1 million by now, as his advocacy for public awareness for global sanitation. He has won so many awards, including the Outstanding UPLB and UP Distinguished Alumnus, among others. Currently, he is a Glenn and Phyllis Futrell Distinguished Professor of Civil, Construction, and Environmental Engineering. Dr. De Los Reyes will discuss the topic on the role of microbes and climate change conversion of waste. Thank you.
Thank you very much, Academician Raimundo, for the detailed introduction of our three first speakers. So I would like to call now on Dr. Jan Leach to present her talk on climate change, impacts on plant microbes interactions.
Good morning and good evening, and thank you very much for the invitation to speak. I'll work on sharing my screen here. Is it shared? Okay, well, thank you very much for the opportunity to speak and to talk to you about how microbes and microbiomes can be part of the solutions to improving agriculture. And thank you, Sean, for that wonderful introduction. As Sean said, her and I go way back. We've been friends for many, many years, and people say we're almost like sisters, except for I'm about a foot taller than Sean, so we look kind of funny walking together. You know, agriculture is facing some really troublesome trends, and these troublesome trends are only exacerbated by climate change. What we're seeing is that the total productivity, which should be following the green line here, this trend, is in fact way lower than it should be. This is 2020 here, and the gap is growing and increasing over time. And this is for the average for the global rates. But if we look at low-income countries, they're falling way behind, which is exacerbating food security. Now, all of this is truly being exacerbated by climate change. And we know in agriculture that climate change will negatively impact agriculture. We see issues such as drought, heat stress in dairy cattle who are particularly susceptible to it. And not only do does it affect their milk production, but it affects weight gain. For other instances, we're seeing flooding and erosion, the opposite effect, drought exacerbation of crop production. We're seeing increased wildfires internationally, and these are seriously impacting not only agriculture, but our atmosphere. But today I'm going to focus mainly on crops, the aspect of agriculture that's related to crop production. We expect and are seeing that with the climate change, we will see reduced crop yields. We already see it in rice production, where a 1 degree temperature over the last 50 to 100 years has dropped rice production in Asia. And we're seeing an increase in disease and pest stress, which is my particular area of interest. So we know that microbes are part of the problem, and that with climate change, we see increases in virulence or mutation to virulence. We can see increases particularly in crop pests in geographic distribution. We can see the range of which they can go because of the changes in climates. They're moving into new ranges. Their dispersal is increased by the weather events. And from the host side, we're seeing increases in host susceptibility as pathogens move into these new ranges. They're affecting the hosts. With the increasing storms that we're experiencing, they're wounding the plants, which is allowing pathogen ingress. And then we're also seeing new movement of vectors, plus also the insect vectors can replicate under conditions, they're increasing replication under higher temperature conditions. So there are many attributes of climate change that are affecting plant disease and its spread and its increase. So we're in fact expecting increased plant disease and stress. But microbes are also part of the solution. There's not a thing about plants that isn't involved, just like humans, that doesn't involve microbe partners. So microbes can support plant health by increasing availability of nutrients, enhancing plant growth, neutralizing toxic compounds in the soil, making plants more resistant to disease, heat, flooding, and drought, which is something I'll talk a little bit more about later, and then also deterring pathogens and predators. Now, in my world, we talk about the need to understand how microbes interact with plants as the phytobiome. And the phytobiome is the interactions of the environment and all living organisms, such as microbes and microbiomes, that influence or are influenced by plants. In our contention is that if microbes are to be part of the solution, we need to understand this entire system, not just the microbes, not just the plants, but how they interact. And that's what I'd like to touch on today. But in order to use that information, there are a lot of knowledge gaps that we need to bridge. And this bridging those knowledge gaps is not a trivial task. And that's because interactions within the phytobiome are super interconnected, meaning that a stress, for example, can alter the metabolism of a plant cell or the physiology of a plant. And they can independently affect the physiology or the biochemistry or the metabolism of a microbe, or they can the effects can be directly on the microbe or the plant, or they can be indirect. For example, if a plant is drought stressed or pathogen stressed, the metabolites that are found in the exudates, or the chemical composition of the exudates from the roots, can change. And we know this to be a fact that when plants are exposed to stresses, they not only exude different chemicals from their roots, but they also have different aerosols, or chemicals that are released from the shoots. But anyway, the changes in these root exudates can impact not only the recruitment or the colonization of microbes from the soil, but it also complicates the interactions of microbes with microbes. So it not only changes how microbes are recruited to the plants and how they colonize them, but how microbes interact with the microbes. These interactions are also dynamic because they depend on the physiology of the plant, the developmental stage of the plant, the timing of the interaction, how the management practices are affecting the plant and the microbes at a given time in a given place, and also soil factors. So that would be an environmental factor such as the moisture content or the pH of the soil. And finally, they're extremely complex. We know that, for example, the communication between the plants and the microbes changes with different kinds of stresses. So it's complicated because you're not exposed to only pathogen stress, but you're likely, if you're standing in a field, exposed to temperature stress, heat stress, or drought stress. So the interaction of a disease stress with a heat stress, for example, may not be the same phenotype or the same response in the plant, or even the same response in the microbial communities as if the stresses were independently applied. So it becomes much more complicated. In order to understand this, we need to not only understand the impacts of a stress on the plant, on the microbes, on the microbiome, the community of the microbes, but on the plant and the microbes and the microbiomes and their interactions with these stresses. So it's very complex. A number of years ago, we wrote a very brief review of how members amongst in the phytobiome would communicate. And normally,
Dr. Leach, your sound is, we can't hear you now.
Is that better?
Yes, that's good.
Oh, sorry. Yeah, it fell out of my ear. So normally when we study these interactions, we study bacteria communications. How do bacteria communicate with one another, such as quorum sensing? And we also talk about how plants communicate with each other with volatiles. But if we think about understanding the whole system, we need to understand not only communication between like organisms, bacteria to bacteria, but also we need to be able to understand inter-kingdom communication. And then what are the outcomes of those communications? So as an example, I'm sure many of you are familiar with quorum sensing in microbes, which is an important tool for a bacterial community to understand how many bacteria are present, and if it's time for them if there's sufficient numbers in that community for them to start signaling, sending out virulence factors, or establishing biofilms, or turning on motility genes. And what happens in some of the interactions with plants is that once the plant senses the bacteria is present, it secretes rosmarinic acid, which is a quorum sensing mimic. And once that occurs, then the rosmarinic acid induces a premature quorum sensing response in the bacteria, which induces the production of virulence factors, biofilms, and other stress response and motility factors in the bacteria. But this is premature because the bacteria really hasn't established enough of a population in order to fully take advantage of their virulence factors. And what happens is with this premature response, they induce plant hormone signaling mimics, but they also impact early activation of plant defense response genes. So instead of causing disease, the bacteria, in fact, activate plant defense, and the plant is protected from the bacteria because it has produced this mimic hormone sensing, quorum sensing compound. So this is an example of interkingdom communication, which is critical to understanding how plants and microbes interact. So part of my point here is that if we want to exploit the interactions between microbes and plants and microbiomes in plants in order to use that to improve crop production or to establish healthy soils or healthy microbial communities, we need to understand the mechanisms by which they interact. And to do this, we need to understand not only how plants sense the stresses, but how microbes sense these stresses. And then how the plants affect the colonization of microbes and how that they also affect the assembly of communities and functions of the microbes, and also how microbes affect plant metabolism and physiology. We need to understand how microbes integrate signaling from stresses in plants, and also how plants integrate signaling and abiotic stresses from microbes. And we also need to understand how different types of stresses affect both plants and microbes. So understanding this system or this complex set of interactions will help us understand how we can manipulate different steps in the process. I'll just give you an example of a very nice study that was done a few years back by Santos and Medellin, where they were studying long-term drought stress in rice. And in this study, what they found is that compared to control, which is well-watered rice, if the rice plants were exposed to long-term drought, they begin to see a change in the microbial community. And what happened is that in that change in the microbial community, the plant root exudates, the chemistry of the root exudates changed, as I mentioned before. The bacteria that were attracted or recruited to those roots were groups of bacteria, including actinobacteria, that were actually beneficial to the plants and actually promoted root growth under drought conditions. So beneficial microbes were recruited under long-term drought. But what was particularly interesting in this study is that when they removed the drought stress, they rewatered the plants, that this group of microbes persisted not only in the rhizosphere, but in the endosphere, inside the plant roots. So this and other studies are leading us to believe that that active recruitment of beneficial microorganisms might be a common evolutionary strategy to enhance plant fitness. So in the coevolution of these organisms together, the plants and the microbes, the plants have learned to attract, using these stress response signals, help beneficial microbes that help the plants grow and that will persist once they're there. But if we're going to take advantage of this response for improving crops or improving the microbiomes around these crops, we need to understand several things. For example, we need to understand how the plants integrate external cues with recruitment of beneficial microbes. We need to understand what controls the persistence of these beneficial microbes after drought recovery. And then if we want to exploit this, we need to think about how we can take these different processes and apply them to improving crops with tolerance to stress. So we need to embrace the complexity of this system. We need to in order to use this knowledge, we need to understand the phytobiomes and then use that knowledge to improve performance in plants. And this is going to require, as I mentioned before, a systems approach. So going forward, we've had a lot of technological advances that have allowed us to make the advances that we've made so far. But there are a lot of things that we still need improvement in order to continue to make advances in these fields. So, for example, we have made tremendous strides in the omics technologies, metagenomics, metatranscriptomics, metaproteomics and proteomics, metabolomics, and in particular, the bioinformatics and statistical analysis that allow us to take that information forward. But when we're talking about sampling from environmental samples, we need to bring down the cost, and we may need to make the throughput of all of these technologies higher and much cheaper. So we need to continue to make advancements in those areas. We need to continue to build our predictive skills, our mathematical modeling skills. They've come made a huge advances in these fields, but there needs to be more continued in those areas. And I'm going to mention more about the advances in culturing and synthetic biology, but we also need, once we get those technologies in place, we need to be able to take these ideas that we have, these innovations, and we need to be able to validate them and evaluate them at scale, meaning field scale. One of the things I want to talk about before I move into a little bit more on that is how we have advanced in culturing microbes from environmental samples. In the last few years, there have been huge improvements in our ability to not only culture organisms from the environment, but to do it in a high throughput way. One of the things that metagenome sequencing has allowed us to do, which is sequencing the entire community, is it's provided insights into microbial function. What are the metabolites that these are producing? What are the biochemical pathways and the physiology of these organisms? That's enabled us to computationally design new culture media to grow some of these recalcitrant organisms from environmental samples. Using genome information, we can engineer antibodies that will be useful for pulling those microbes or attaching to those microbes so that we can isolate them from environmental samples and enrich them and help us to study them independently. And also we can use gene-targeted cultivation. We can target media that's actually towards particular genes. And the other part of the culturing is that we're seeing new techniques and mechanisms that enable high throughput culturing. And that allows us to isolate many of these environmental microbes and study their especially their interesting traits, like new antibiotic productions and different things like that. But one of the more exciting tools, I think, that is going to allow us to finally break into understanding how bacteria interact within a bacterial community, within a microbiome, is the in situ genome engineering that has recently been developed. It's still a very new technology, and it's not for every lab at this point in time, but it was developed out of Jennifer Doudna's lab, where you can actually genome edit a microbe in a community and then monitor, that allows you to understand how that function of that microbe fits into its ability to colonize or to live within a community of organisms. And this is particularly important as we want to develop synthetic communities of microbes. We need to understand who will live with who and who will provide important functions for our community going forward. So just to sort of tie all of that together, I've described here a potential pathway that one might use for the development of a synthetic community or a SynCom to apply to a smart farming system. So for example, this walks you through everything from sample sites where you're comparing high performing with low performing varieties or disease suppressive soils, characterizing those with all of the omic technologies available, selecting particular isolates based on their function, isolates that are important within the core isolates and the hub isolates, the ability to culture those isolates and manipulate them and understand the traits that they produce, perhaps alter those microbes using the in situ genome mutagenesis or genome editing that I mentioned, and then applying all of this information using mathematical modeling, integrating all this information so that you can begin to predict what microbes might be the best microbes for in a synthetic community, which ones will live together, which ones will coexist and provide the beneficial traits to the plants or to the soils, and then we can move them into the fields for evaluation, where again we need to have high throughput tools for monitoring the impacts of these communities on the plants. So moving forward, we need to embrace the complexity of this system. We need to be able to determine the makeup and the functional potential of plant-associated microbiomes and microbes to unravel the complexities of interspecies and ecological interactions and metabolic networks. Importantly, we need to move beyond correlation to causation. We need to get to the mechanisms, because that's how we're going to be able to apply this information in the future if we understand what to manipulate or what to change. We need to be able to model and predict. So we need huge advances in the ability to monitor host genotype by the interactions with the microbiome genome, the environment, and the management practices in order to tailor solutions that will be long lasting and be appropriate under a changing climate. We need to develop and adopt standardized procedures for collecting, classifying, and reporting consistent and well annotated data. This is something that has been a need not only in the age of genomics or metabolomics or proteomics. This was needed even in the age of trait and physiological and biochemical traits of microbes. We need to develop globally accepted standards for plant-associated microbial products in order to ensure that once we have developed them, that we can translate them into real-world conditions. And most importantly, I think we need to train a next generation of collaborative phytobiome scientists with skills in integration and the ability to share best practices across the many disciplines that it takes to address this complex problem. So microbes are part of the solution for adapting agriculture to climate change. And we care about this because we can identify new genes and pathways and possibly new compounds that are applicable in agriculture. We can also develop new strategies for disease control, such as synthetic microbial communities. Instead of trying to apply one microbe, we can apply a community that's that's developed to live together and to work together. And importantly, we want to understand the system to avoid mistakes, because we don't want to, for example, develop heat-tolerant plants that are more susceptible to the microbes that are in the community, or develop microbial mixtures where the important steps, the important processes that are beneficial to the plants are there, but those microbes can't tolerate one another, or they can't live together. So hopefully with all of this integrated knowledge, we'll be able to avoid those mistakes. I just want to mention very briefly a new project that I've become involved in recently, which is to understand the aerobiome, or what's happening in the atmosphere. And this is important from the point of view of what microbes are in the air and moving in the air for human health and for plant health, for agriculture and animal health. And so what we're asking is, what microbes are living in the air in communities, or are they in communities? How do they get there? What influences their composition? What functions do they need to stay there? How do they get there? And finally, what are they doing there? Are they interacting with one another? Are they nucleating ice? Are they exchanging genetic material? Or are they just simply being transported in the air? So this is a new NSF-funded project that we have underway that is actually quite exciting. We're looking at the aerobiome over fire systems, over agricultural systems, and in plant and animal agriculture. So I hope I've convinced you that embracing the complexity is not only necessary, but that it is an exciting way forward in understanding how microbes influence plants under climate change conditions. Thank you.
Thank you so much, Dr. Leach. Please give a hand to Dr. Leach. That was really fascinating, comprehensive and clear overview on plant microbe interactions. And you presented a lot of exciting pathways in research in this area. And it's a complex but fascinating system for sure. And it's actually converging, whether you're talking about terrestrial or marine ecosystems. So very, very exciting. So thank you again. So for just a reminder, the questions will be, there will be an open forum later. So please remember to key in your questions in the Q&A box, and also for those who have access in the webinar chat. So Dr. Leach, please, we will have questions later for you, but thank you. And for now though, we will be moving on to the next part of our program. So to discuss the microbes in climate change, adaptation and mitigation measures in agriculture, we call now Dr. Noel G. Sabino.
Thank you very much, Dr. Iniguez, and also to Ma'am Raimundo for the kind introduction. So please allow me to share my screen. Okay, can you see my screen now? Okay, and can you hear me properly?
Yes.
Okay, so magandang umaga po sa ating lahat. Okay, good morning to all of us. Okay, now for today, I'm going to discuss microbes and climate change, and that is the role of microbes, specifically the soil microorganisms, as well as adaptation and mitigation measures in agriculture. Now, of course, warming of the climate system is unequivocal as stated by the IPCC. Now, Earth's temperature has changed by a rate of around 0.08 degrees Celsius since 1880s. And as mentioned earlier by Dr. Leach, now this is something to do with anthropogenic production of greenhouse gases. And as mentioned by Tej, climate change is considered as the most serious contemporary challenge facing humanity. What are the impacts of climate change? Of course, we have warmer temperatures, so we are now experiencing it. And due to warmer temperatures, you now have increased frequency, intensity, as well as duration of heat waves. Now, worsening air and water quality, changes in the patterns and amount of rainfall, of course, affecting agriculture. Now, increases in the frequency and intensity of extreme weather events, such as heat waves, droughts, and floods, rising sea level, which is affecting coastal ecosystems and communities, changing ecosystems influencing geographic ranges of many plant and animal species, thereby affecting their migration as well as reproduction. Now, it is said that climate change is happening globally, but the effects differ locally. Certain regions, as well as human populations, are more vulnerable to the impacts of climate change. Now, climate change is affecting all sectors, including that of agriculture. Extended droughts, excessive precipitation patterns, emergence of new pests and diseases, as well as flooding, is affecting soil fertility as well as productivity of agricultural ecosystems. As such, different strategies were developed in response to the impacts of climate change. Now, considering such strategies, they can actually be classified into two. Of course, you have mitigation, that is intervention to reduce the sources or enhance the sinks of greenhouse gases, as well as adaptation, that is adjustment to actual or expected climate and its effects. Now, with the need increased production, okay, that is crop productivity, okay, in order to support the increasing population, okay, the Food and Agriculture Organization of the United Nations in 2010 okay, introduced the concept of climate smart agriculture, which is an integrative approach to develop agricultural strategies for sustainably increasing agricultural productivity, adapting and building resilience in agriculture and food security systems, and reducing, of course, the impact of agriculture to climate change. Now, with the importance of microorganisms in the agricultural sector, okay, interest is increasing with regard to the harnessing of soil microbial communities to mitigate the negative consequences of climate change. Now, soil microbial communities are very important, and that has something to do with the ecosystem services that they provide. That is provisioning, supporting, as well as regulating. Now, it is said that climate change is one of the most important ecosystem services provided by the microorganisms. Now, the soil microorganisms, such as the bacteria and fungi, okay, they actually play an important role in land-atmosphere carbon exchange. Now, microorganisms play a very important role or a key role in climate change because they are agents of biogeochemical cycling, and they are capable of producing and consuming the three dominant gases responsible for 98% of the increased warming. Carbon dioxide coming from respiration as well as microbial decomposition. Okay. Nitrous oxide coming from the activity of ammonia oxidizing bacteria as well as denitrification. And of course, you have methane due to methanogenic microorganisms. Now, soil is a major concern in climate change because of their ability to regulate soil organic carbon. It is said that the soil stores around 3.1 times 10 to the 5 kilograms carbon, which is more than 2/3 of the total terrestrial carbon. If these organisms will mineralize soil organic carbon, and this will contribute substantially to greenhouse gas emissions. Now, of course, it is important to determine how the microorganisms will respond to climate change. Now, it is reported that climate change pressures alter the soil microbial community structure and functional processes. For example, elevated carbon dioxide present in the atmosphere is said to cause an increase in below ground carbon, that is the carbon present in the soil, affecting soil diversity as well as function. It also leads to increase in soil organic carbon. But in the long run, due to priming effect, there will be a drop in soil organic carbon. Seawater intrusion, increased precipitation, so other impacts of climate change. They also promote anaerobic conditions in the soil, which is conducive for methane synthesis, as for methane production, as well as nitrous oxide emission. Now, what are the mitigation or adaptation strategies which can be used in order to manipulate soil microbiome? Now, knowing the response of microorganisms to the different impacts, we are now able to manipulate the soil microbiome to mitigate and adapt to the negative consequences of climate change. There is a direct as well as several indirect methods which are being used in order to manipulate the soil microbiome. One such method, which is commonly being used, is of course the use of plant inoculants. Now, this is a direct way of altering the microbiome. Plant inoculants are available commercially. They may be made up of bacteria or fungi or a combination of both. And usually these organisms come from the rhizosphere. Now, these organisms play an important role in agricultural stress management and ameliorate the negative impacts of climate change. Drought, extended periods of drought, erratic precipitation patterns. Now, inoculants are important and they can be used for addressing such impacts. Bacteria which are capable of producing extrapolymeric substances, for example, can be inoculated into the soil, that is to increase water retention. Plant growth promoting microorganisms which are capable of producing phytohormones such as indole acetic acid can be used to stimulate root formation, and that is to alleviate water stress. Now, in the work of Itakura et al., they were able to show that the use of an inoculant can mitigate nitrous oxide emission. Using Bradyrhizobum japonicum with an active nitrous oxide reductase activity, they were able to show a significant reduction in post-harvest nitrous oxide emission. Nodule decomposition said to be the reason why in post-harvest conditions, nitrous oxide is being produced in the soil. Degradation of nodules will lead to the release of ammonia, and through nitrification as well as denitrification, nitrous oxide is produced. Unless the microorganism possesses the nitrous oxide reductase, it will not be able to convert nitrous oxide to nitrogen. Now, with the use of inoculants, that is positive or is capable of synthesizing the nitrous oxide oxidase, reductase, and that is actively functioning nitrous oxide reductase, it is able to mitigate nitrous oxide emission that is at the field level. Indirect methods were mentioned earlier as means of manipulating soil microorganisms. Now, these are actually traditional methods which are being used by farmers, and it is only now that we are showing that they are very important in climate change mitigation. One such procedure is conservation tillage. It is any tillage practice that builds up crop residues on the soil surface to minimize the impact of water and wind erosion. Conservation tillage, such as, of course, reduce, minimize, or zero tillage, direct drilling, as well as strip cropping, can actually enhance soil aggregation, promote biological activity, and increase water holding capacity, as well as infiltration in the rates in the soil, leading to greater available soil moisture, improved soil tilth, and increased organic matter content. The work of Al-Hassan, they were able to show that no tillage, as well as no tillage together with straw, is able not only to increase grain yield, but also to mitigate emissions of greenhouse gases. Okay? Looking at the table, it can be observed that net carbon dioxide flux, methane, nitrous oxide emission, okay, as well as global warming potential and the greenhouse gas intensity were reduced significantly when no tillage or no tillage with straw were used. Another common practice of farmers is cover cropping. Now, cover cropping is growing any non-cash crop in addition to the primary cash crop. It is said that cover cropping improves soil health, enhance water infiltration, smother weeds, control pests and diseases, as well as increase biodiversity. It should be noted, however, that the nutrients which will be introduced into the soil and the microorganisms or the diversity of microorganisms as well as their functions will be affected or is affected by the type of cover crop which is commonly being used. Okay. Now, leguminous crops, okay, such as Sesbania, Cetosema, were reported to be used as cover crops, and they were shown to increase microbial abundance and diversity. And being leguminous in nature, okay, they increase nitrogen present in the soil, and at the same time improve nitrogen cycling, carbon cycling, as well as phosphorus cycling. In the work of Busia et al., that is making use of vetch, that is a nitrogen-fixing legume, they were able to show that is through chloroform treatment, that is chloroform fumigation, incubation approach, that there was an increase in the microbial biomass carbon in the soil. Now, the same is true was observed with the microbial biomass nitrogen. They were significantly increased, okay, as compared to that of the other treatments. Using profiles, that is fatty acid methyl esters, they were able to show that cover cropping also influenced soil diversity. And that is, it stimulated the growth of gram-positive bacteria as well as actinomycetes. Now, it is said that these organisms are suited in the so-called environment where a lot of aromatic compounds are present. Okay? These organisms are capable of utilizing a lot of recalcitrant substances. Another land management practice which is being used to mitigate the impacts of climate change is agroforestry. Now, agroforestry is actually an intentional integration of trees or shrubs into agricultural systems. Now, similar with cover cropping, the type of vegetation being used, okay, will definitely have an impact on microbial diversity as well as function. Now, soil microbial biomass as well as microbial diversity are observed to be greater due to the ameliorative effects of trees and organic matter inputs, as well as the differences in litter quality, quantity, and root exudates. Now, there are several types of agroforestry which are being practiced. And it is, of course, dependent on where the crops are planted in relation with the trees. We have alley cropping, that is placing or planting crops between trees. We also have forest farming, that is a forest protecting, okay, the crops. We also have silvopasture. Now, in the case of silvopasture, you are integrating livestock into the farm. We also have windbreaks as well as riparian forest buffers. Now, shown in the table is the so-called carbon sequestration potential of different agroforestry types of the types of agroforestry. Now, using different crops, as mentioned earlier, definitely will have a different impact, okay, on the microorganisms present, as well as on the soil carbon, which will be incorporated. Okay? Looking at silvopastoral system, that is using Acacia mangium and Arachis pintoi, that is with the age of 10 to 16 years, okay, and a depth of 0 to 100 centimeters, there is a concentration of about 173 milligrams soil carbon per hectare. Now, comparing it with another silvopastoral system, now this time using a different set of plants and trees, okay, that is of the same year or age, of the same depth, okay, there was a lower concentration of soil carbon, indicating that, of course, the type of crops being used will definitely have an impact on the microorganisms as well as on, okay, the properties of the soil, leading to, of course, accumulation of a certain amount of soil carbon. A very common land management practice is soil amendment, okay? And that is products are being added to the soil to enhance desired characteristics such as nutrient content, texture, and fertility. Now, two soil amendments which are said to improve not only crop production but at the same time mitigate greenhouse gas emission are biochar and compost. Now, biochar is a carbon-rich product from pyrolysis of biomass such as wood, crop residues, and manure, while compost is managed decomposition of organic materials by organisms under high temperatures. Now, what are the benefits of biochar and compost? Okay? It is said that these amendments can improve soil health and water quality. And of course, this will increase crop yield as well as reduce the need for synthetic fertilizers. And as mentioned, they are capable of reducing greenhouse gas emissions. Production of compost and biochar. Now, if we consider compost production, now please take note, compost production is actually a source of greenhouse gases. That is carbon dioxide, methane, as well as nitrous oxide. And as such, the procedure being used in composting are constantly being modified in order to reduce greenhouse gas emissions. Now, zeolite, which is an aluminum silicon crystals, which is an absorbent, can be incorporated into the manure. Now, this is true for manure compost in order to reduce methane as well as nitrous oxide oxidation. Now, some researchers are using biological additives in order to mitigate greenhouse production or emission. Use of nitrite oxidizing bacteria will definitely cause a drop in nitrous oxide being produced. Superphosphate or monocalcium phosphate has also been tested in composting, and it was shown that it can substantially cause a reduction in greenhouse gas emissions. Now, in the case of biochar production, it is said that these materials are heated, that is at a very high temperature under low oxygen conditions in order to produce the biochar. Now, both the compost and the biochar can be added directly into the soil, separately, or compost and biochar can be combined as one. Now, what are the benefits observed from the use of biochar? Now, we look first at the so-called properties of biochar. It is highly porous, it has a large surface area, and of course, several functional groups, as well as alkaline pH. Now, because of these properties, it is able to strengthen aeration, that is in the soil, when it is introduced into the soil because of the pores, providing air into the soil. It is of high absorption, because of the so-called surface properties of biochar. And of course, you have to consider the different functional groups which are present. It also improves microbial activity. In addition, it also provides venue or sites where the microorganisms can attach to, and also it improves pH. Now, in fact, it is said that biochar is highly effective, especially in poorly irrigated soils, or that is with soils which can easily be drained, such as sandy soils, and it can improve the fertility of such type of soils. Now, biochar, with all its properties, it is capable of absorbing, of course, greenhouse gases, specifically carbon dioxide, methane, nitrous oxide, and also ammonia. Now, in the study which was conducted by Yang involving the use of biochar, they were able to show that the use of biochar was increased not only grain yield, but it also caused a drop in greenhouse gas emissions. Total carbon dioxide emission, that is methane, nitrous oxide emission, as well as the global warming potential and the greenhouse gas intensity of the gases were reduced significantly. Now, as for composting, of course, following the modifications in the procedure of composting to reduce emission of greenhouse gases, the benefits obtained from composting include, of course, increasing soil organic matter, increasing NPK, that is no need for other fertilizers, increase micronutrients, increase microbial activity, reduce soil-borne diseases, increase water holding capacity, increase cation exchange capacity, reduce soil erosion, reduce soil compaction because of the formation of organic matter, which will now form soil aggregates, as well as increase carbon sequestration. Now with that, what are the key points of this talk? One, soil microorganisms play a key role in climate change, serving as producers and consumers of greenhouse gases, of course, depending on the condition. The impacts of climate change also affect the microorganisms. But please take note that the soil is a very heterogeneous environment. That is, the nutrients are patchy, some areas are anoxic, others are said to be aerobic. So definitely different responses will be exhibited by the microorganisms as affected by the soil properties. As such, it is suggested, or it's a good thing to do for scientists working on this field to collaborate and share information in order to come up with a true picture of how the microorganisms respond to the impacts of climate change that is coming from soils of different environments and property. Now, direct and indirect manipulation of soil microbiome can be done in order to mitigate and adapt the impacts of climate change. Now, as mentioned earlier, looking at the so-called strategies which are being used in climate-smart agriculture, these are actually traditional practices which are being used by the farmers. So I think it is our duty to properly inform about the so-called benefits of these traditional practices, and that is through intensive information education campaign. And finally, first, we need to make policy making bodies to consider the importance of soil microorganisms in climate change management, particularly in the agriculture sector, because of the important role that they play in this sector. With that, I would like to thank you for listening.
Thank you so much, Dr. Sabinos. Please give him a hand. Thank you for providing us with an idea on the various approaches on adaptation and mitigation measures, particularly for the soil microbiome and the impacts in agriculture. So as a reminder, again, we have an open forum where we will be discussing the questions that you've submitted. So at the end of all the talks, so please do type in your questions in the Q&A box if you haven't done so. And also for the Facebook Live participants, please put in your questions in the comment section there. So thank you again, Dr. Sabino. For now, we will be moving on to our next speaker. So to give a talk on the role of microbes in climate change, conversion of waste, let us listen to Dr. Francis L. de los Reyes III.
Thank you. And good morning, everyone, and good evening for those of you in other locations. So this is really a big topic in terms of conversion of wastes and the role of microorganisms. And I chose to focus on a few key things. And so I look forward to the discussion also. See if I can get this going. Okay, so this is the I guess the classic figure that shows all of or a systemic view of microorganisms in the environment, in space, and also changing in time. And in particular, what we want to do is we want to take a look at the role of microorganisms in waste. And so I'd like to highlight this part here at the lower right, where you're looking at microbial decomposition, microorganisms performing respiration, performing methanogenesis. So this is happening in nature. And really what we're doing in environmental engineering, in microbial ecology, looking at waste conversion is really harnessing the same processes, but maybe in a faster way in more controlled environments. So I just wanted to put that in context. So we'll focus on this. So if you look at the global greenhouse gas emissions, this is a graph from 2016. Looking at the different sectors that contribute to GHG emissions. You can see energy is the big one here, 73%. Energy used in industry, energy in transport, all the cars that we use, all the buildings that we occupy, commercial and residential, as well as fugitive emissions and fuel combustion. All of these is a huge part of emissions of greenhouse gases. The next big sector is agriculture, forestry, and land use, and Noel has started really talking about this. And then you have industry also. But I wanted to focus on this portion here on waste, which is 3.2%. And really here in this graphic, it's broken down into landfills and wastewater. So I wanted to focus on that 3.2%, which you might say it's a small number, but 3.2% of a big number, annual emissions of 50 billion tons of equivalent CO2 is also a big number. Okay. So all of you go to the toilet, all of us produce wastewater, and you can do these rough calculations. So for example, you have 7 billion people in the world, and roughly on average, about 0.25, 0.3 pounds of BOD or organic matter per person per day. And if you use a conversion factor of about 0.5 kilograms CO2 per kilogram COD or VOD, you can see that there's billions of pounds of CO2 released every single day, right? Potentially, if you convert all of that organic matter that we produce. Now, obviously, that's really part of a short-term carbon cycle because that BOD or that organic matter, that carbon is coming from the food that we eat. But you can imagine that depending on how you draw the boundaries, these calculations become quite, based really on context. All of that wastewater that we flush ideally should go to wastewater treatment plants. And this is where a conversion of that organic matter happens. And really what we're doing is we're converting pollutants in wastewater, in water, and we're converting that to pollutants in the air. So for example, the carbon goes to CO2, the nitrogen goes, the ammonia, becomes nitrogen gas, for example. And that's really what's happening. So that's why wastewater treatment is contributing to climate change. For example, in an aerobic process like activated sludge, what you're doing is you're really feeding the microorganisms, and these are heterotrophs that use the carbon, and they're aerobic heterotrophs, so that they use the oxygen and they produce carbon dioxide and more microbial biomass. This process produces biosolids, and these biosolids are typically also stabilized. An example would be with anaerobic digesters. And here, those organics are converted to fatty acids and then eventually to methane and CO2. And we know that methane is another very potent greenhouse gas. Another aspect of waste is the landfill, right? So solid waste formation. And again, a properly designed landfill would be an anaerobic process where essentially you have a lining, you have a leachate collection system, it's capped, and then you have a biogas collection system that you can flare or even collect and use for energy. Again, it's an anaerobic process. The solid waste is converted to methane and CO2. And the numbers are just going to keep increasing. This is from the World Bank data of projected waste generation in tons per year in different regions. And you can see that the biggest changes are going to happen in East Asia and the Pacific. That's really China, right? From 2016, which is the green bar, to 2030, the yellow bar, to 2050, the orange bar. South Asia is the next big region that also has this huge change to 2050, and that's because of essentially India, right? And then sub-Saharan Africa. In the next 20 years, you're going to get an increase in solid waste production from sub-Saharan Africa. So all of this has to actually be taken care of. We actually have to think about where this solid waste is going. I mentioned that in a landfill or in an anaerobic digester, the microbial processes are anaerobic. And this is really the classic anaerobic food web. You start with complex biopolymers, whether they're proteins, carbohydrates, or lipids. They're fermented by fermenting bacteria or fermenters, and broken down into monomers, simple sugars, amino acids, and so on. Those are further fermented to short-chain fatty acids like propionate and butyrate. And in the process, you produce hydrogen and CO2. You produce acetate. There's a pathway from the short-chain fatty acids to acetate, and these are the acetogens that do that. So these are all your bacteria. And then from acetate, you have archaea that basically split the acetic acid, acetoclastic methanogens that produce methane and CO2. There are also methanogens that reduce the CO2 and use the hydrogen as the electron donor and make methane. So this is basically the pathway by which in an anaerobic system, you get your organics becoming methane. And again, in our context, we're concerned about this because methane is a potent greenhouse gas. Now, in my lab and in many labs, we look at the specific microorganisms that are involved in the degradation of specific substrate types. Here's an example of a network based on 16S rRNA gene sequencing, where you can look at different substrates. So these are the different colors, and then the starting microbiomes, and figure out which microorganisms really are doing the work. And so this is just a quick snapshot of the different kinds of microorganisms that might come up depending on the type of substrate. So for example, if you have lipid-based substrates like fats and oils and grease, on the methanogen side, you're going to see an increase in methanocyanida, the syntrophic bacteria that convert fatty acids to acetate, Syntrophomonas, for example, and so on. So identifying the active organisms and the functioning microorganisms actually provides a way of understanding how to maximize a reactor system, a digester, so that you get the maximum conversion to methane. And then obviously, assuming that you capture the methane, you can use that for energy. Here's an example of a graph where on the x-axis is the organic loading rate of fats, oil, and grease waste. And so the higher the number here, the more you're loading your reactor. On the y-axis is the methane yield in liters of methane per gram of volatile solids added. And what I'm showing here in the different dots and circles and triangles are the different literature values for methane yield. And so what you want to do is have a higher methane yield on the y-axis. And the triangles and the squares are actually from our experiments. And to date, we have the highest levels of methane yield. And we were able to do this by actually figuring out the microorganisms that we needed to make sure are dominant in these reactors and cultivating them through a series of stresses of the right food and the right step increases. So this is a whole study, and I'm just showing the ultimate result is that we can get to very high levels to maximize our methane potential. And this again is for fats, oil, and grease waste. The other big kind of waste that most people don't really talk about, but I'm sure people have heard about, is food waste. Globally, we waste a lot of food. You can debate the numbers, but it's about 1.4 billion tons of food waste each year. If you look at a typical dump truck and the volume of a dump truck, it's equivalent to lining up dump trucks around the earth 27 times. And that's how much food waste is lost each year. And so it's lost all along the process, right? So you've heard numbers, maybe, for example, 40% of all food actually does not make it or is not eaten. So that's the definition of food waste. And it's lost in the production side. On the left-hand side, you have agriculture, in the processing, and then in the restaurants, institutions, in retails and households. And the size of the food icons here actually are relative to the size of the food waste in each step, right? So people have done analysis of step by step where the food waste is coming from. And here's an amazing statistic that essentially, if you think about food production, so let's look at this long bar here at the bottom in gray. Food production is actually responsible for 26% of global greenhouse gas emissions. So this includes agriculture, right? It includes cultivation, it includes, farming and so on. But because the food eaten is only a fraction of that, if you do the math, actually 6% is lost, consumer waste and loss in supply chain. So really, food waste accounts for 6% of total global greenhouse gas emissions. It's a huge, huge number, and something that obviously we can do something about. So going back to landfills and food waste that may go to landfills and food waste that may be anaerobically digested, these are numbers from the top 5 emitting countries, and these are all in metric tons of equivalent CO2. So you can see the US is number 1, and then China, and then Russia, and then Mexico. And then interestingly, it's really interesting that Malaysia is the 5th largest country there. And by 2030, the projection is that landfills, non-CO2 greenhouse gas emissions, so mostly methane, landfills will account for 7% of all of those non-CO2 emissions. Obviously, energy is still there, and agriculture, and so on. So what can we do about this? So the US EPA has this graphic that talks about different types of technologies that can reduce methane emissions from municipal solid waste landfills. And so the bars here talk about the reductions achievable, and the bright blue to the line here are reductions that are actually can be achieved at very, very low cost, right? And then the other reductions, as the blue becomes more faint, actually that means that reductions are becoming more expensive. So let's take a look at some of the technologies. If you actually capture the methane and you generate electricity with this reciprocating engine, that's the most bang for your buck, right? You can actually reduce up to 40% of methane emissions. Waste to energy is another thing. So essentially, you could even just burn the solid waste. And then mechanical biological treatment, composting, anaerobic digestion, which we had just mentioned, and capturing the methane, recycling paper, because paper is one of the largest components of municipal solid waste. Recovering the landfill gas for direct use. And that may be for something like, directly using the gas that you recovered. Even flaring of landfill gas is better than just letting the methane go out. In other words, if you burn the methane after you collect them, you're converting the methane back to CO2, and that's better than just letting the methane actually leave the environment. Enhanced oxidation here just means, part of this is using, for example, methylotrific or methanotrophic microorganisms, for example, that may convert your methane to CO2. So you've got these microorganisms that are actually that use methane as electron donor, oxygen as electron acceptor, and convert methane to CO2. And then electricity generation depending on the technology. And so if you think about all of these, you can reduce 12% of the methane emissions right away without any real cost. But then if you want an additional 49%, then the cost keeps increasing, but it's still technically feasible. So, these are some of the projections of the US EPA. What about for wastewater systems? So similarly, these are the top 5 countries, but instead of, now you have Russia as number 1. This is now in terms of wastewater and methane. And then India, China, and then India, and then Mexico, and then Nigeria, and then finally the US. And in 2030, wastewater methane emissions are expected to be about 5% of total non-CO2 emissions. Now, similarly, we can ask, what can we do to limit methane emissions? Here are some, right? So convert a latrine, we'll talk about that in a little bit, to aerobic wastewater treatment, convert an open sewer to aerobic wastewater treatment, and septic tank to aerobic wastewater treatment. And then using a treatment plant with an anaerobic sludge digester, capturing the methane again for energy. And according to the EPA, at no cost at all, we can reduce by 1%. We can reduce up to 35% of methane emissions using various technologies, but they would actually have increasing cost. Now, we've talked about CO2, we've talked about methane, but actually in wastewater, another big pollutant is nitrogen. What I'm showing here are the different types of nitrogen transformations. The classic ones are ammonia oxidation. So ammonium in the wastewater is oxidized to nitrite and then to nitrate by ammonia oxidizing bacteria and nitrite oxidizing bacteria. But there are other types of transformations. So the classic denitrification from nitrate, stepwise, all the way to nitrogen gas. There's also denitritation, nitrite going through NO to N2O to N2, and then NMOX, anaerobic ammonia oxidation, where organisms like Brocadia take ammonium and nitrite and then convert that to N2. Now, I'm pointing this out because these pathways actually produce N2O. So nitrous oxides are also a very potent greenhouse gas, about 300 times the impact of CO2. And so depending on how you do your nitrogen removal in your wastewater treatment plant, you're going to get some of these N2O. So for example, this is one study that looked at the global warming impact and the quantities of methane and N2O in wastewater treatment plants. And you can see the numbers, and N2O actually has a bigger impact. And this is normalized per cubic meter of wastewater. Okay, and this is by direct measurement. By direct measurement, by simulation, you can see that N2O is really the bigger problem than methane. Now, we've talked about microorganisms and their role in specific wastewater treatment processes. But I want to point out that you can think of this as systems of systems. And so if you look at the very inside box here with this brown box in the middle, that's what we call within the fence or inside the wastewater treatment plant. And again, you can get your emissions in methane and N2O and CO2, like we talked about. If you have, again, anamox, for example, or partial nitration, you're going to get N2O. If you have anaerobic digestion, you're going to get methane, and so on. But as you increase the box, you might have infrastructure that's related to wastewater treatment. The sewer line, for example, you have emissions. The trucking of food waste, you have emissions. If you think about the transportation, right, the burning of fossil fuels for fuel, to get your truck going. The ultimate disposal of your biomass in landfills or incineration, you get all of these emissions. And then finally, you can even have a bigger box, this bigger outside blue box, where you think about all of the inputs, chemical production, electricity production, and so on, and the effluent going into a river, going to agriculture, all of the emissions associated with all of that. We can also count that essentially as part of how we think about wastewater, right? We can do the same kind of analysis for landfill leachate for solid waste. Okay. What this is really what people talk about as scope 1, scope 2, scope 3. So scope 1 is the direct emissions coming from the processes. And then scope 2 would be the indirect emissions from the electricity that we buy or the heat that we buy. And then finally, scope 3 are the emissions upstream and downstream because of all of the infrastructure, materials, chemicals, and so on that are involved in wastewater treatment. So as we increase the viewpoint or the lens by which we view greenhouse gas emissions in wastewater treatment, you can see that really if you start counting, the impact is much more than just what's inside the treatment plant. And this is another graph that I wanted to show, and this is really changing it over time, right? So the x-axis is the year. This is data from China, from paper by Li et al. And you can see everything increasing in terms of equivalent CO2. The blue space here is really the CO2 because of the electricity needed. The biological CO2 is the red one, and so it's a little bit smaller than the electricity that's needed. And so you can start really accounting for all of the greenhouse gas emissions. And I know we're talking about microorganisms here, so we're interested in the biological portion of this. But really, the biology will not happen without the inputs of energy, right? And so you have to think about the reactor also and the system. Now, not everyone uses centralized wastewater treatment. This is a figure from the IPCC, and this is how they actually kind of look at emission factors and inventories of greenhouse gas emissions from waste. The chapters are here. If you look at volume 5 and volume 4 of the IPCC reports on wastes and greenhouse gas emissions, you can see how they handle quantifying greenhouse gas emissions. I wanted to put this up to show you that there's all kinds of things like uncollected wastewater. And even when you collect the wastewater, you have a variety of technologies available to you. Billions of people actually don't have toilets at home that safely manage their wastes. And some, like 1.9 billion, don't have basic sanitation services. And in other words, what we call an acceptable toilet facility. And about 500 million people still defecate in the open. And so for these folks, they don't have a centralized wastewater treatment system. They have all kinds of technologies, maybe a hanging latrine like this, right? And how do we then account for greenhouse gas emissions and the waste conversion of microorganisms in these scenarios? And one way to think about that is the sanitation service chain. So basically, you're looking at the toilet to containment, conveyance, treatment, all the way to reuse and disposal. So there was a study that just came out from Kampala, Uganda, where they tried to look at the greenhouse gas emissions for the different technologies, the different approaches for sanitation, and line pit, septic tanks, and so on. The emission factors are here: conversion to methane and then conversion to nitrous oxide. What they found was quite interesting. They found that if you look at the numbers here, majority actually is in the treatment. A lot of the greenhouse gas emissions actually are in the actual treatment of wastewater and also in the containment, even on the direct methane and direct N2O emissions. In other words, it's really in the sanitation chain, it's the treatment of wastewater, at least in this particular city, that's important. You can quantify which technologies contribute to the greenhouse gases. And it's interesting that open defecation, pooping in open ground actually does not give you a lot, right? Because it's an aerobic process. Essentially, the organic matter is converted to CO2 and not to methane. So the greenhouse gas total equivalent CO2 is a little bit lower. But then you have all of these other things, other technologies that actually give you a lot more emissions. And you can also take a look at the ultimate fate of the fecal material and quantify the CO2 equivalent. And again, if the highest is actually delivered to treatment, treated, and discharged, which is really, really interesting. So what do we do about this? I think the solution is really looking at a circular economy where we reuse our products, we convert things to energy so that we can mitigate a little bit the impacts of the conversion of waste to greenhouse gases. So this is an example circular economy diagram where you can have your wastewater and your water system and your energy system more integrated, so that you can actually think about how you can recycle chemicals, for example, in the system. So, for example, fecal sludge can be made into all these different products: soil conditioner, protein, biofuels, and we actually have some research on biofuels and making energy from fecal sludge. Here are some examples of what we can do. We could get biogas, we can get syngas, we can get biochar, as was discussed earlier, and bio oils and alcohols and so on. So these are all technically feasible. We can convert fecal sludge, for example, to protein. And here's an example. This is the black soldier fly. The larva of the black soldier fly eats the waste. And then what you do is you harvest the larva. And then you can dry that and make that as a protein supplement or substitute for example, fish meal or soybean oil meal in animal feed. So that's one way you can actually use the organic matter in fecal sludge into something that is more valuable. Okay, so what do we need? I'll end with this, some research needs. We need to have systems-based analysis beyond the traditional boundaries of just the individual processes. We need long-term research data observatories that can really look at methane generation from septic systems and esteros and wastewater treatment plants in the Philippines. We need the data that's based in the Philippines and models that can be accessed by different researchers, because this affects not just waste, but other things in food and water. And finally, highlighting circular economy research and projects. This can be biofuels using microorganisms. This can be conversion to energy using microorganisms. How do we highlight those, and how do we make that really part of policy going forward? And with that, I look forward to the discussion later. Thank you.
Thank you very much, Dr. De Los Reyes. That was very insightful and provides us a broader idea of waste and how complex and interactive these systems are. So again, please do put your questions for Dr. De Los Reyes in the Q&A box or for the Facebook live participants in the comment section. So, and we will tackle those in the after this last presentation. So, with that, we will move on to our last, but not least, speaker. And I will first introduce our last speaker, who is Dr. Deo Florence Onda. So he's an associate professor and also the deputy director for research at my institute, the UP Marine Science Institute. So his research interests includes understanding host-symbiont interactions, microbial biogeography, diversity, dynamics, and trophic interactions, and consequences of changing conditions using various omics approaches, other molecular methods, and advanced techniques in confocal laser scanning microscopy. So he received his inter-university PhD in oceanography, specifically microbial oceanography, at the Laval University in Quebec, in Canada, and his postdoctoral fellowship at the Alfred Wegener Institute in Germany. So may we please call on Dr. Onda to deliver his talk on the need for long-term monitoring initiatives to understand climate change effects in the Philippines. Deo, you have the floor.
Thank you, Ellen. Thank you for that very generous introduction. And I'd like to thank again the organizers for the invitation. I think I'm also presenting a very unique perspective relative to the previous speakers, because I think the experience of early career researchers also open up opportunities for other young researchers in the country. So I hope to also share that on what we can do relative to the issue of climate change. So let me just share my slide. So while actually listening to the other speakers, I thought of modifying a bit the presentation to also align and also integrate some of the discussions that have been made in the previous presentations. So for this presentation, and thank you to Dr. De Los Reyes for initially discussing already the need for long-term monitoring initiatives. And I'd like to focus on this need, particularly in the Philippines, to understand climate change effects. So I just would like to emphasize the slide that you're seeing right now is actually showing the research vessel, the first research vessel of the Marine Science Institute. And later, I'll be able to share to you why having these kinds of platforms is actually very significant in the context of the Philippines. In 2019, we've already seen this slide earlier. Scientists actually gave a warning to humanity, and it emphasized the role of microorganisms in climate change. This slide has already been explained to you earlier, but I just would like to emphasize what they actually said in that particular paper, that the impact of climate change will depend heavily on the responses of microorganisms, which are essential for achieving an environmentally sustainable future. However, being a microbial ecologist myself, the challenge really for us is to understand the mechanistic processes. We always call this as the black box. We know that things go into the ocean or they go into a certain part of the environment, and those things actually get transformed or converted into something else. As discussed by Dr. De Los Reyes, for example, we've got wastes that turn into gases or greenhouse gases in particular, but some of the mechanisms involved are still not yet known. And that really makes us difficult to understand how are we going to mitigate probably, how are we going to manage and come up with long-term solutions to these problems. So we call this a black box because we know that there's something getting into the box, something is going out of the box, but we actually don't know the mechanistic processes. And that is a key information for us, again, to actually tackle the issue of climate change. And one way to understand that black box is to come up with long-term data sets, long-term monitoring efforts, and long-term initiatives. And why is it that long-term monitoring systems are important? Because long-term data will provide comprehensive baseline data across time and space, we'll be able to understand how the environment is actually changing based on the indicators that we're looking at, relative to time and relative to different spaces. It also allows us to actually monitor changes both in the environment and the biological patterns or the biological communities. There are certain changes in the environment that cannot be detected by snapshot data, meaning that gathering data once a year or every once in a while will not actually allow us to really detect the cumulative minute changes. So having long-term monitoring systems will provide information and capability to detect cumulative effects of long-term processes, including climate change-related events. Long-term monitoring systems are also important because it will allow us to come up with bases in establishing regulatory thresholds and limits, for certain pollutants or even activities. And globally, there had been an effort to actually establish many long-term ecological research stations. So there's the International Long-Term Ecological Research Station Network and the Global Ocean Observation System that allowed different researchers from the different parts of the world to actually harmonize or standardize their methods of collection and also share this data with each other and come up with a more holistic, more long-term, high-resolution insights into the changes that are happening in the environment. So this global map actually shows you where those ecological research stations relative to the marine environment are actually located. And I just would like to emphasize the green circle that you're seeing is the Philippines, and there's none in the Philippines. There is no long-term ecological network, ecological monitoring station that is located in the Philippines. And this is very important for us because the Philippines itself, in terms of oceanography, is a confluence of the different oceanographic processes. And therefore, as stated earlier, we might even be one of the most vulnerable countries relative to climate change. One of the manifestations of climate-related changes or events is really the changing sea surface temperature. The sea surface are getting warmer, so this is a data from NOAA showing you how the more red it is, the warmer it becomes, and through the years, the waters in the Pacific, including those part of the Philippines, have also increased in temperature. These global changes have been suggested to result in different or have implications to biological patterns in which and their distribution, which include, for example, species migration going northward or going southward, where it is actually less warmer, or species displacement, which will then affect ecosystem functioning, and some organisms that cannot adapt to these changes might actually be lost, then therefore resulting to biodiversity loss and the loss of the ecological services associated with that biodiversity. Now, the question is that how can long-term data sets actually help us understand the consequences of these changes? So one example that I'd like to give to you is a very I think unique and also a classical example is what's happening in the Arctic. So I've done my research, I've done a PhD in the Arctic, and one of the interesting things is that we know, for example, that the Gulf Stream, which is a warm water coming from the tropics, enters the Arctic through the Fram Strait, which is between Greenland and Norway. Now, the thing with the Fram Strait is that there is a unique distribution of a certain type of species or ecotype that is adapted to very unique conditions, which is the organism Micromonas. Micromonas is an important global phytoplankton. However, within that particular species of Micromonas, there are what you call the cold type species, which are limited in the north or limited in very cold regions of the world, and warm type species that are very much limited in the tropics, in the temperate region. Now, once this warm type species actually crosses the Fram Strait, they don't survive because the conditions are not favorable to their growth and proliferation. Now, with climate change, there is a very distinct event that is happening in the Arctic, and that is the melting of the ice because of the warming of the sea surface. Now, with the melting of the ice as an indicator of climate change, the question is what happens to species distribution of these very unique ecotypes? The Fram Strait is very unique also because the cold water coming from the Arctic goes down through the coast of Greenland, and the other current that goes up into the Arctic that brings the warmer water, or the West Spitsbergen current, is actually bringing the species as well as the warm waters from the tropics. There is a long-term monitoring station found in this area. It's called the Hausgarten, that has been established by Germany and has been collecting data since early 90s. Now, there is a very unique event that happened in the Arctic. They call it the warm anomaly of 2005 and 2007. During this warm anomaly that coincided also with the highest sea surface temperature in the area and the greatest ice melt that occurred in the Arctic, they observed that there's warmer sea surface, there's a decrease in ice extent, and there's also lower productivity. So by having this Hausgarten, they were able to actually detect these significant events. Now, the question is that how do these climate-related changes could have changed the distribution of different species? In the Arctic, again, there's a warm cold and a cold type species. So by actually detecting the presence and the abundance of this species, we were able to observe that the cold type species, or those that are more adapted to the colder conditions, actually decreased in abundance in warmer years. And then the warm type species coming from the tropics and the temperate actually increased their abundance significantly in warmer years in the Hausgarten and also in the Arctic. Not only that, we also observed that some species, such as, for example, Emiliana, that are more warm adapted, have actually increased their abundance, and cold adapted types of phytoplankton, such as Phaeocystis, decreased in abundance. So this actually provides us with some evidence of Atlantification process, a process where the Arctic is now becoming like the Atlantic because of the changing conditions and because of the changing temperature regimes. This could result in species displacement, loss in biodiversity, and changes in the ecological functions in the Arctic region. Now, why is it actually significant as an example for the Philippines? Because the presence of the ecotypes of Micromonas and other related species are not only found in those two extreme environments. There's this species called the Osteococcus. Osteococcus is a very small phytoplankton, but also widely distributed globally, but have been identified to have specific niches, meaning that they have specific distribution. For example, you have Osteococcus that are only found in the Mediterranean, certain Osteococcus that are only found in temperate regions, And then while doing research in the West Philippine Sea, but their distribution has not yet been really described or explored in the West Philippine Sea and the temperate in the tropical waters of the South China Sea. But doing research in the West Philippine Sea, our studies actually provided some evidence that there is a very unique clade of Osteococcus that is only found in the west of the Philippines. We call this the West Philippine Sea clade, which might actually be very much adapted to the conditions that we have in the West Philippine Sea or in the tropical waters of the South China Sea. Now, what is the importance of actually describing or identifying that species? Because Osteococcus, that particular West Philippine Sea clade of Osteococcus, might actually be used as an indicator on understanding how is the waters in the Philippines actually changing. And this is relevant because questions have been asked. If species are getting displaced or actually migrating north or south of the tropics, the question is what happens to the Philippines? This is still a very relevant question that many scientists, including Dr. Iniguez, I think, are still trying to answer in the Philippines. And we're still looking for good models on how to answer those ecological questions. So identifying the diversity of the phytoplankton, actually identifying very unique ecotypes found in the West Philippine Sea, can be used to actually answer these types of climate-related questions. Another approach that we can use in order to understand how the waters as well as the species of in the Philippines might be adapting or how will they survive in future ocean conditions relative to climate change is through forecasting or modeling. One way to estimate how climate change will affect marine microbes is through ecological niche models. So ecological niche models, there are different types of it, which are mechanistic, correlative, and also a hybrid of those two. The more simpler one are the correlative models, which can estimate habitat suitability and probability of occurrence through known environmental conditions where the taxon is present. So if you know the current tolerance or range of conditions that current species are thriving in, then we might be able to predict how will they survive in future ocean conditions. However, in order to come up with very good niche models, we need data. We need, for example, occurrence data, their presence, their absence, their background. And then we need to understand where did they come from? Where were these observations reported? Aside from that, if we have the data for the biological entities or organisms, we also need data from the environment. We need to look at the temperature, the salinity, and other relevant environmental parameters where these organisms actually survive. So my group and one of my students, Ms. Jana Calpito, has been trying to come up with ecological niche model for the Philippines. We're trying to find a model organism. And we thought, starting with the diatoms, which are widely distributed all over the Philippines, might be easier primarily because it is easily detected. It is reported widely, and therefore the data to actually come up with a model for diatom would be easier. But actually, it's not. So the Philippines is also important to emphasize that when we do niche models, we need to understand that the Philippines is responding differently relative to climate change. The different biogeographic regions of the country, for example, the West Philippine Sea, the North Pacific Seaboard, the South Pacific Seaboard, Sulu Sea, Celebes Sea, Visayan Sea, are responding differently. And therefore, to understand really the effect in the Philippines, we also need to understand the different biogeographic and oceanographic regions of the country. David et al., for example, has reported that the different parts of the Philippines will be affected differently, with ocean acidification being more prominent in the north, extreme heating in the west, extreme rainfall events in the southwest, and also sea level rise in the east. Therefore, understanding should also include considerations on the different responses that the different biogeographic regions in the country might actually experience. Now, going back to the niche modeling exercise that we've been doing, this is the problem that we had. Although many data are available for some parts of the Philippines, including the West Philippine Sea, there's still really a lack of data in many biogeographic regions, including Sulu Sea, and most importantly, there's none for Celebes Sea. At least this review of literature or data was just based on published articles or published papers and reports. We do not have access for those that are unpublished, so they were not included in the analysis. So it really emphasizes in what part of the country research should be done and more effort should actually be concentrated. Aside from that, when we tried to understand and tried to use the available data, or at least the available publications, we observed that majority of diatom occurrences or reports of diatoms in the Philippines, as reported from literature, actually lack minimum information for correlative ecological niche modeling. For the publications that we reviewed from the 1980s to 2021, we observed that 74%, imagine that, 74% of these publications actually have no exact coordinates for georeferencing. We cannot actually place them in the map just because the reports do not include information on the location. 42% of that 74% actually have no coordinates and no other in situ environmental data. They just reported that there were diatoms, but we cannot locate where they are, and they do not have environmental information. 130 or 57% have no coordinates, but with other in situ environmental data. So relative to data gathering, information collation, and synthesis, I think there's still also a gap in that. It is difficult even to look back into the previous data because the information are very much limited and lacking. So that's one challenge, no? But I would like to emphasize that there are certain groups of organisms where data have been a bit more available. This is a work done by our moderator today, Dr. Iniguez, and colleagues from the other Southeast Asian countries. And this is just really a demonstration on how long-term data can actually be used in order to look at effects, for example, of long-term processes such as the El Nino oscillation. to understand possibly climate change. But even if we are already seeing a very good 2-decade or 3-decade data set here, they also emphasize in that particular paper that there's still a need to enhance long-term monitoring and establishing hub species distribution across the region. So even though there is already an effort right now, we actually need to improve those initiatives and efforts. So what do we need to do? We need to come up with widespread and systematic sampling. We need to also standardize or even just harmonize our reporting so that other researchers can make use of them and compare and use them in their own scientific efforts. We need to come up with there's no repository of data collated in the Philippines. There are individual and separate efforts to collate this data, but they are not publicly available. And there's none that can be found in any of the government agency websites or repositories. Now, we also need to understand that climate change can also be exacerbated by the other events that are happening, other problems and other pollutions that are happening. For example, eutrophication, which can exacerbate HABs conditions, or the other way around, climate change being exacerbated by HABs. And also plastics pollution, which is an emerging important issue in Southeast Asia. So one of the efforts of our research group is to come up with a comprehensive collation, integration, visualization, and publicly accessible data set for plastic pollution in the Philippines. So this project is funded by the UST PSHERD, and it is now publicly available. You can visit the website. It's called plasticount.ph, which is already the most comprehensive collation of all pollution surveys done in the Philippines. Now, why I'm actually and you will notice from this particular website that there's not much data available in the country. There are certain data where it is much accessible, for example, in Palawan and also Manila Bay, but majority or many parts of the country still do not have enough data for plastics pollution. Why am I showing this to you? Because when we talk about plastics pollution, the role of microbes is actually very important. Microorganisms are the first groups of organisms that encounter the plastics and the first that colonize the substrate. And therefore, they also have the control and influence on the fate of the plastics itself. For example, microbes that form biofilms can actually attach to it and then be transported via plastics. Later on, biofouling as well as formation of biofilm can actually assist or facilitate in the deterioration and therefore in the creation of microplastics. Microplastics, when they are coated by biofilm, they can be mistaken as food by smaller animals such as zooplankton and larvae of fish, and therefore can be passed on to the higher trophic level, assisting or facilitating biomagnification. Later on, there are also reports of enrichment of microbes that can degrade or decompose the plastics, which then contribute to the production of carbon dioxide and other greenhouse gases. So therefore, plastics might actually even contribute or exacerbate the ongoing climate change scenario. Some of the results that we have, for example, is this, no? We saw that there are a lot of bacteria present in the environment, naturally occurring consortia that can inhabit and then degrade plastics. This is a publication of one of my students, Justin Bitalak, and the microscopy image that you are seeing on the right are actually bacterial colonies that are degrading or decomposing plastics. And with this decomposition, again, is the possible production of more greenhouse gases just because plastic itself was derived from fossil fuel. The other thing that we are very much worried about also is that plastics, when they are coated with biofilm, they can actually also host other organisms. Not all organisms are the organisms that we want. Our studies, for example, in Manila Bay have shown that many of the biofilms you find in plastics are also serving as home or habitats for toxic species such as harmful algal bloom forming species. You've got Alexandrium, Protopyridinium, and Scripsiella. And we've shown in the lab that the biofilm can actually help this HAB species survive long transport. So imagine if there are so many plastics going out of Manila Bay and all of these plastics have biofilm with many HAB species attached to it. And these plastics go somewhere else, Batangas, even in the remote islands of the West Philippine Sea. It actually helps species migration. The cells alone cannot travel or cannot cross the ocean. But if they are attached to something where they have access to nutrients, then it will facilitate their wider dispersal, allowing then the proliferation of microorganisms or these dangerous hitchhikers in other parts of the country. And with climate change, those two working together might even increase the occurrence and distribution of hab species. This also emphasizes, again, the need that answering climate change problem should be integrative and should be cross and transdisciplinary. There are now actually a move to go towards more integrative approaches. For example, using omics and using oceanographic information, combining them both to actually come up with more insights into climate change scenarios. And this is what we've been doing in our research group, trying to make use of omics and high throughput sequencing approaches in order to understand the mechanistic processes, that particular black box, to understand how climate change is actually happening and how organisms are responding to it. However, in the context of the Philippines, we have been lagging behind in adopting these technologies. It was only in 2018 when the first paper using high throughput sequencing was published. It was, the number of papers coming out using this particular approaches are still a bit low, but we have started adopting it, and hopefully there will be more young researchers who would take on the challenge of actually integrating these different approaches. I would like also to emphasize, as an early career researcher in the Philippines, the pursuit for science should be complemented by investments in infrastructure, in platforms, in funding opportunities, and most importantly, people. At the end of the day, it is the researchers who stay in the lab, who go out into the field and analyze this data. So we really need to invest in our young people. So I just would like also to share, there are some movements happening on the ground. For example, in our research group, we are implementing this big consortium of 10 different state universities and colleges across the country, funded by CHED. We call it the Habs Watch Project. And relative to this, we have already established initially a Philippine Ocean Observation and Monitoring Network. So the map that you're showing right now are the current monitoring stations of the program. We're trying to capture the different biogeographic regions in the West Philippine Sea with our collaborators from Luzon and Palawan, in Sulu Sea with our collaborators from in Zamboanga and Palawan too, in Visayan Sea with our collaborators from Visayas, and also in Southern Tagalog, and also in Mindanao and Celebes Sea. The first time that we will be seeing data coming out of Celebes Sea. But we still don't have a collaborator from the North Pacific Seaboard in northeastern Philippines. So if you're interested, just let us know. But that also emphasizes really the limitation in terms of expertise. It's difficult to find collaborators, primarily because these types of work are not mainstream. They are not the most interesting, but we need them in order to understand what's happening in our waters. So this Philippine Ocean Observation and Monitoring Network simultaneously conducts sampling and monitoring across major biogeographic regions. We're now using and integrating high throughput and integrative approaches, such as, for example, the use of omics, imaging flow cytobots, and even machine learning or artificial intelligence to have better data analytics, and also making the data publicly accessible. So we will be launching an app called the Habs Watch app, hopefully in the coming months, where people can actually go there and see what's happening in the monitoring station, also download the data for their own use. I think another big move and another big development in the marine scientific community and marine environmental research right now is the establishment of the National Academic Research Fleet. Before, I've shown you the photo of the only vessel that the Marine Science Institute has right now. But we are very happy to share to you that we are now building a fleet of different vessels. We are in the process of procuring 3 more vessels, large vessels that can go into the open oceans. These vessels, including Panata of UPMSI, will be deployed and dedicated to the different biogeographic regions of the country, including the West Philippine Sea, the Northeast Philippines, the Sulu Sea, and also the Visayan Sea. So hopefully with these platforms coming in, it will also make younger scientists become more interested to do oceanographic research and contribute in collecting data to answer societal problems such as climate change. I think another important milestone that we had in the field of marine environmental research in the Philippines is the establishment and launching of the Pag-asa Island Research Station. We know that the Pag-asa Island is in the middle of the South China Sea, making it actually a very strategic place to establish a monitoring station away from the coast, away probably from the effects of pollution coming from the coastal environment, and then therefore making it easier to tackle and to really separate the effects of climate and other long-term processes. So this Pag-asa Island Research Station in Kalayaan Island Group was officially relaunched in October 2022 and has become fully operational last year. We've already deployed researchers there for a few months, for a couple of months, and we are now talking with the national government to actually make this open to more researchers, including international researchers. We always believe that although Pag-asa is located in a very controversial, where tension on geopolitical issues is actually high, we believe that science can be a jumping point where we can enjoin other countries to go back to the negotiation table and talk about marine environmental conservation and protection. So if this is Pag-asa, the monitoring station or the Pag-asa Island Research Station is actually here. So hopefully for the other researchers listening today, you might want to actually already consider, in your future proposals, going to Pag-asa, coordinating with us so that you can do your own research in Pag-asa Island. So hopefully with those examples that I've shown you and the efforts that we are trying to implement right now, long-term ecological data are important because they allow us to look back into the past. They also allow us to understand what's happening right now, the current events. But more importantly, long-term monitoring data will also allow us to look forward into the future, allowing us to actually become more prepared of the changes that are happening in our world. So I hope you've learned something, one or two, and thank you very much for the opportunity. Salamat.
Okay. Thanks, Dr. Onda. So thanks for presenting that view of microbial oceanography from the Arctic all the way from the Arctic here to the tropics, and also, of course, for emphasizing the definitive need for long-term ocean observation initiatives if you really want to tackle climate change and the local issues that are occurring even as we speak. So with that, we have a lot of food for thought from all the speakers. So thank you very much again to our four speakers, Dr. Leach, Dr. Sabino, Dr. De Los Reyes, and Dr. Onda for your many insights. And to our participants for listening and hopefully learning a lot. So now we would actually first, we will now proceed to the open forum. And there's a lot of questions from our participants. So we think we will, first, some of those have the panel have already answered, but we might want to still open them so that the rest of the larger group of participants can listen and also contribute. So first question that came in was from actually academician Rasco for Dr. Leach first. So the question is, plant breeding has traditionally focused on resistances to harmful microbes. Do you see a future for plant breeding that aims to develop traits that confer plants the ability to attract friendly microbes? Second, considering the complexity of microbial systems, do you see the need for co-culturing instead of pure culture? So Dr. Leach, maybe we can share for the larger audience our thoughts on this.
Sure. Thank you for the question. Yes, indeed. One of the things that microbiome scientists are working towards is, can we identify the attractants and the needs for a community to establish around a root, and the beneficial traits that those microbes might provide for the plants. So again, it's a complex system. They need to understand what genes in the plant are important for producing the attractants and not producing repellents. And then also need to understand what traits the microbes have that might be beneficial to the plants, and understand how the microbes fit into a community and can establish as a community. But yes, indeed, that's exactly what we're working towards, is identifying the genes that the breeders can move into plants that will help attract microbes that are beneficial to the plants. And your question on co-cultivation is important because especially environmental microbes sometimes are not easy. Obviously, many are not easy to culture. And it may be that we need to co-culture them in order to even culture them at all. It may be like in some of the cell culture systems where you have nursicultures to help provide the nutrients or provide something that we don't know what is to get something to grow. So yes, that's a big possibility and an area of research.
Thank you very much, Dr. Leach, for answering that question. Maybe we can go around. Actually, there's a related question for Dr. Leach as well, is in terms of the plant's ability to sense approaching stressful activity in the phytobiome. It's somehow related to the human defense system. Would it be possible to manipulate this response of plants to successfully program their defense mechanism?
And again, that's another big area of research, understanding innate immunity in plants and how we can adapt plants so that they are better able to resist pathogens. And first of all, be better able to detect their presence and then better able to resist them. And at the same time, we have to be careful, because if we prevent a pathogenic microbe from colonizing the plant, we may also be preventing a beneficial microbe from colonizing the plants. So that's when I say we have to be careful about making mistakes. That's one of the things we have to be careful of.
Most definitely. Yeah, actually, I wonder with the synthetic biology approach, where do you think would be the kind of the key, like how to safely proceed in that area while avoiding those kinds of mistakes that could be very costly?
Yeah, I think that may be the benefit of the genome editing, is that it's a very minor shift rather than a major shift like our traditional mutagenesis practices. So yeah, I think everything has its risks, and we have to understand what those risks are and how well we can get around them.
Thank you, Dr. Leach.
Mm-hmm.
Maybe some questions moving to Dr. Sabina. I mean, the rest of the panel, of course, are free to also input their thoughts and for any questions as well. So please just chime in if you wish to do so. So specifically for address to Dr. Sabino, the farming practices, especially in rice fields, may be considered as a source or a sink of carbon and greenhouse gases. Every planting cycle, the rice fields are cleared completely through burning of the dried husks or the rice straws. Would this practice means as a biochar input to improve the soil condition of the plantation area to enhance the greenhouse gas absorption of soils, or will this contribute to emissions rather than mitigation?
Thank you very much for the question. Now, please take note, burning of rice straw should not be done because you're actually releasing greenhouse gases. Okay, and at the same time, if it is being done for quite a long time, you're actually losing nitrogen, phosphorus, and sulfur from your field. So, and of course, we also have the law which is now banning burning of rice straws. Okay, and please take note that is not considered as biochar, because when you produce biochar, it is under oxygen limiting conditions or oxygen deficient conditions. Now, burning, okay, in the field, actually you have a lot of oxygen, so this is being converted to carbon dioxide. So definitely you are contributing more to greenhouse gases rather than mitigating emissions. Okay.
Thank you. That's a good clarification for regarding this question of the system. So there's also further question for you, Dr. Sabino. Microbial inoculants have that generic problem of lacking applicability across soil environments. So what steps are being explored to solve this problem?
Okay, so that is one of the so-called disadvantages of using inoculants. Now remember, inoculants are actually isolated and they are grown under laboratory conditions. So when you apply them in the field, they will be exposed to various factors, and this will definitely, make it difficult for them to thrive, that is to compete with existing populations. So I think for the use of inoculants, you have to consider a certain organism which is capable to withstand a lot of environmental conditions so that it will be able to grow in the environment where you are to inoculate it. Now, usually what is being done is if you want to inoculate the organism, it should come from the environment where you are to introduce it so that it is already already acclimatized in the so-called environment. Like in one of the so-called studies, that is the use of bradyrhizobium. The so-called bradyrhizobium was taken from the soil. It was screened in order to get a certain quality, a certain important trait, and then it is again brought back into the said environment. So it's very difficult to come up with an inoculant. Very rare or very little percentage will you have an inoculant which will actually work in the field, with a certain, let's say, success rate as that when it is grown under laboratory conditions.
Thank you, Dr. Sabino. I think, that's a very similar issue as well with any kind of microbial approach to issues in the ocean as well, where you have to consider different heterogeneous environments and also connections of the water to other areas. So you have to be very careful with applications in these kinds of these approaches. So would anyone want to contribute more to that question? Are we good with that? No. There's actually a lot of questions. So there's more for Dr. Yes, Dr. De Los.
Yeah, maybe I can add to that a little bit. I think Dr. Sabino gave a really comprehensive answer. Just placing it in the context of microbial community assembly. So adding, outsiders, inoculants or bioaugmentation is really called immigration, right? So these are immigrants to the system. And if you look at the bioaugmentation literature, it's a hit and miss affair. So there are a lot of companies that sell, bioaugmentation type organisms and formulations, and a lot of them make money. We've done studies side by side in full scale systems, and these are waste treatment systems where basically there's no effect, right? And you can look at the actual causes, because if you think about how much you're adding versus how much is already in there, just cell counts or biomass, it's really not going to be, make a difference if you're adding less than 0.01%. So that's one thing, right? But then there are some success stories. For example, in groundwater contamination, it's possible that just adding the right microorganisms, like, the halococoides that can fully dechlorinate, PCE and other chlorinated substances, then, sometimes they do report successes. So it's a hit and miss affair. And the factors that go into that are under this microbial community assembly theory that I'm also interested in as a research topic.
Thank you, Dr. De Los Reyes. And yeah, so that's that speaks to really understanding that black box that they was graphically mentioned in the mechanisms promoting those interactions and succession in the assemblages in these kinds of systems. I think actually, Dr. De Los Reyes, so there's a question for you. So Academician Rasco Jr. asked, Bill Gates advocates a decentralized waste management system in the toilet. Do you believe this is a viable idea? What is the impact on climate change, and will it work in poor households?
Yeah, so actually, I'm a big believer in context-based solutions, and that means that not everybody has access to a sewer, and it's not feasible, not cost-effective for everyone to be flushing their toilets with clean drinking water, and then connected to a sewer, miles and miles of pipes that are basically using up a lot of energy and a lot of water. So yes, we actually have research and a lot of work on looking at optimizing between centralized and decentralized or distributed systems. There are lots of issues with that, right? So you can think about decentralized systems as household level. You can have your own household scale, waste and water recycling system, for example, separating your gray water from your yellow water or urine water from your toilet water, shower water, and so on, right? So you can think about treating on-site and reusing some of that water. At the household level, you can do that. And then at a cluster or community level, you can also get away with smaller systems. So sometimes those make sense, right? So the issue with a lot of these distributed systems is always the operation. Meaning, you don't have the economy of scale to have skilled labor, to have, operation and maintenance people coming if something goes wrong, right? So the hope is that with new technologies like sensors and wireless devices, that you can actually overcome that disadvantage. And so the idea is you have a lot of sensors and with Wi-Fi, you can monitor a lot of small systems from a centralized location. Then you are also maximizing the economy of scale in terms of the operation. So there are lots of research and thoughts and ideas in this, but I think a lot of the world is thinking this way now, that decentralized systems might be able to serve, especially underserved communities everywhere, around the world.
Thank you, Dr. De Los Reyes. It's actually heartening to hear that kind of those kinds of systems that could be more feasible in many areas. Right? So we have also questions for Dr. Onda. We have, can we expect an increase in microbial diversity as a consequence to ice melting in the polar caps when ancient microbial life, which has been frozen in time, are being revived?
In terms of increase or decrease, that would be very difficult to estimate. But we know that there are already evidence of biodiversity loss, meaning there are certain particular groups that cannot survive without the ice. But it is also true, and reports have shown that there are certain types of viruses that have not existed in current times that are starting to be detected, which have been, recognized to also be very primitive, which means that they've been probably buried there for thousands of years. So there is a shift in terms of biodiversity. Will this actually have a net result of increase or decrease? That we don't know yet. It's still a question, as also discussed by Dr. Leach earlier, that what would be the implications of that particular change in the structure, in the composition? Because definitely that will have implications on the functioning of the ecosystem itself, and then therefore on the ecological services. And I think that's also one of the, in the field of health, that is also becoming a field of concern, because there are certain viruses that are being, released in the permafrosts that could also be causing diseases or illnesses, and we are not adapted to those kinds of. We did not co-evolve with those types of diseases. So how are we humans, and also the current state of the different animals will adapt to those types of new diseases. So there's still a lot of question on there. But it also opens up a lot of opportunities. And that's what I see. As a young researcher, it just means that there's more work to do, and therefore there are more opportunities for young researchers like me. Yeah.
Yeah. So you have a lot of work to do. No. Okay, so actually there's another question for you that can be more directly linked to also the waste aspect. So can the plastic degrading bacteria that you've observed or isolated be used now in fermentation type system for degrading plastics?
Yeah. So this is the fascinating thing about the biodegrading bacteria relative to plastics. It's been reported globally. There are already evidence that it is happening, since different indicators have shown that the plastic is really degrading or being decomposed. But the problem is we have not identified all the possible genes that are related to the biodegradation. There's one, the peptase gene, that was described in Japan, but other than that, we were only finding proteins that might actually be involved, but we have not really fully elucidated the genes and even characterized and really demonstrate that these genes are directly involved in the pathway of degradation. I also would like to recap what the previous researchers mentioned earlier, that there's no one bacteria that will do the work. What we are finding right now is that it's really the consortium. One bacteria, because they're very specific, they will target one particular backbone, and another group of bacteria will actually attack the byproducts of that. So there are two concepts here. One, trying to understand which genes are involved, and second is what are the different taxa that need to work together in order to really fully degrade the material. And there is now a consensus that we should move towards the consortium type of approach. We need to identify first what types of bacteria can and who can work together in order to degrade. Can now it actually be used? I always say that this is still a long game. We have seen indications of biodegradation, but the processes are really, really slow. So the other challenge is to identify what conditions will trigger or increase the rate of the decomposition or the production of those enzymes. But unless we've already identified the enzymes, it would be difficult to optimize the condition. So it's really a full cycle. There's still a lot of things that we need to do, but at least initial insights have already given us on what direction to go. So yeah, I'm not banking on it yet. It's still going to be a lot of work, but we are, I think, in the right direction right now.
Okay. Thank you, Dr. Onda. So definitely, with all the presentations and from this discussion, there's many exciting avenues to be explored in the complex world of the microbes, and it's really, like, to me, it's really cool that we've I think that so many people, and there's so much more awareness about this invisible world that used to be, pretty much discounted. So little by little, it's their huge impact is a lot more recognized in our current situation. So I don't know if we still have time for questions for the organizers, or is it time for the synthesis? Think. Okay. So we do have to finalize this open forum and the webinar. We did go a bit over time, but it was a lot of very insightful presentations and great discussions and questions. So for this last part, again, thank you to our speakers. And for now, we want to take, of course, the most important part. No, I'm just kidding. To take our photos. So we please may request everyone to turn on their cameras for a group photo. The NAS Secretariat will be taking the photograph. So please let us know, NAS organizers, if you are going to take our picture and when it's finished already.
I'll count to 3 po. 1, 2, 3. One more. 1, 2, 3. Thank you po.
Thank you. So for the final stretch, I would like to call on Dr. Charina Gracia Banay to deliver the synthesis of the discussions. Dr. Banay is a professor of microbial ecology and the current head of the Environmental Biology Division of the Institute of Biological Sciences in UPLB. She's an affiliate professor of the Faculty of Management and Development Studies at the UP Open University and a diplomate of the Philippine Academy of Microbiology. She is also a reviewing editor for the Microbiology Spectrum, a scientific journal published by the American Society for Microbiology, and a section editor of the Journal of Agricultural Research, published by the University of Southern Mindanao. She obtained her BS Biology major in microbiology in 94 and MS Microbiology in 2000 and her PhD Microbiology in 2011. She's been the recipient of several institutional and national awards, and recently the Hitachi Global Foundation Asia Innovation Award together with her advisor and mentor. Dr. Banay has been studying various aspects of food systems, including the role of fungal endosymbionts in bioremediation and in mitigating the effects of climate change in the agricultural setting. So Dr. Banay, please present the synthesis. So thank you. I think you're still muted. Okay.
Hello, good morning, everyone, and thank you for the kind introduction. So we have listened to 4 distinguished speakers on the topic of microbes and climate change in terrestrial and marine environments. It has been an enriching, engaging, and enlightening 2 hours. In the first talk, Dr. Jan Leach discussed climate change, microbes, and agriculture. She started with the observed trends in agricultural productivity that are really quite troublesome, especially since climate change will exacerbate the negative trends. Dr. Leach discussed that while microbes are part of the problem, they are also part of the solution. As climate change intensifies the occurrence of plant diseases, microbes present a viable solution by virtue of their intimate association with plants in various life processes. In particular, she emphasized the highly complex and dynamic interactions of the phytobiome with its host plant and with each other. And these are valuable for climate change adaptation. But to exploit the phytobiome, a mechanistic understanding of the interactions is imperative. There are many knowledge gaps that require in-depth studies, such as interkingdom communication, stress physiology and signaling, and beneficial microbe recruitment and persistence, to name a few. Understanding phytobiomes and using that knowledge for plant adaptation to climate change requires a systems approach. Dr. Leach ended her talk with some recommendations for future research and development, namely advances in enabling technologies, such as new microbial culture technologies, syncoms, and targeted gene editing, for example. Ecological studies on phytome plant-host interactions, mechanistic studies, modeling and prediction, developing standardized procedures, and global standards for microbial products. And lastly, training the next generation of scientists. In the second talk, Dr. Noel Sabino discussed the various climate change adaptation and mitigation measures in agriculture. He started with the impacts of climate change on agriculture and how climate-smart agriculture can help in climate change adaptation and mitigation, specifically in harnessing soil microbial communities for this purpose. Soil microorganisms provide important regulating, supporting, and provisioning ecosystem services that form the basis for their beneficial effects. Although soil microorganisms contribute substantially to greenhouse gas emissions, and are in turn affected by climate change, they can also be harnessed to support various mitigation and adaptation strategies, such as the use of beneficial plant inoculants. Various land management practices that promote soil health also affect microbial communities. Soil amendments, such as compost and biochar, and management practices that promote healthy soil and increase microbial diversity and activity, either directly or indirectly, can help toward climate change adaptation and mitigation. Therefore, policymaking bodies should consider the importance of soil microorganisms and promote soil health in climate-smart agriculture. In the third talk, Dr. Francis de los Reyes discussed the roles of microbes in waste conversion and how this impacts climate change. He started with a systems view of microbial processes and focusing on microbial decomposition as a source of greenhouse gas emissions. In particular, wastewater treatment plants convert BOD and solids to carbon dioxide and methane. Landfills also release methane and carbon dioxide. Greenhouse gas emissions from waste will increase as global waste generation is expected to increase in the next few decades. Identifying the specific organisms involved in the degradation of different substrate types is important to maximize the degradation of wastes and convert them to energy. Food wastes are also a huge source of greenhouse gases, but the greenhouse gas emissions reduction is feasible using various abatement measures and technologies. Greenhouse gas emissions from wastes have up to 49% reduction potential. Aerobic wastewater treatment plants with anaerobic digesters can help further increase the reduction potential. But a more holistic view of greenhouse gas emissions from waste shows that the potential impact of reducing greenhouse gas from the whole system is actually much larger. What can be done? The solution is a circular economy, particularly integrating conversions of waste into value products such as soil conditioner, protein, reclaimed water, biofuels, fodder, and building materials. Finally, Dr. de los Reyes identified 4 research needs, namely systems-based analysis of greenhouse gas emissions, long-term microbial ecological research observatories, Philippines-based data and models, and circular economy research and projects. In the last presentation, Dr. Deo Onda discussed how long-term marine environmental monitoring helps to understand climate change effects in the Philippines. He started with the challenge of the black box, which is unraveling mechanisms. Towards this end, long-term monitoring systems are important to provide comprehensive baseline data, monitor changes, detect cumulative effects, and establish thresholds. In the Philippines, there is no long-term marine monitoring station, even though we are one of the countries most vulnerable to climate change. Long-term monitoring of microbial marine species that can be used as indicators of environmental changes can help in detecting and predicting climate change effects. Ecological niche modeling also helps to estimate how climate change will affect the marine ecosystem in different ways. Modeling can help in teasing out how different biogeographic regions are affected by the various occurrences related to climate change. In this case, adequate georeferencing and in situ environmental data are important. Dr. Onda mentioned the need for widespread and systematic sampling, standardized reporting, and establishment of an accessible repository of collated data. In addition, marine microbes are subjected to effects of multiple factors that interact with climate change. Thus, the need for a wider long-term monitoring of various interacting factors other than climate change indicators, such as microplastics pollution, degradation, and dispersal. There is also a need for integrated methodological approaches, moving towards more transdisciplinary approaches to understand mechanistic processes. But scientific endeavor should be complemented by investments in infrastructure, platforms, funding opportunities, and people. Finally, Dr. Onda emphasized collaboration and networking, as well as high-throughput processes to come up with comprehensive data to understand global concerns. Long-term ecological data allows us to look back into the past, understand current events, and look forward into the future, providing us adequate preparation for the changes that are coming. In summary, all the four presentations showed the important role that microbes play in climate change. While microorganisms contribute to climate change, they also help in understanding, adapting to, and mitigating the effects of climate change. Microorganisms are major drivers of biogeochemical cycles, and they provide invaluable ecosystem services. They are both producers and consumers of greenhouse gases. It is imperative that we study how microorganisms affect and are affected by climate change to fully understand how to adapt and mitigate its effects. The applications of microbes are wide and far-reaching, as we saw from the four presentations this morning. And their study can provide us with invaluable insights on climate change and its cascading effects. Long-term studies, mechanistic studies, collaboration, interdisciplinary research, more enabling technologies, standardized protocols, modeling, and increased investments in research are some of the common recommendations for going forward. There is also a need for promoting innovations for microbe-driven climate change solutions that support human well-being and environmental health. There is a long way to go. We recognize that. There are many knowledge gaps, as illustrated in all of the presentations this morning. But we are called upon to rise to the challenge, embrace the complexity of the interactions within and between various systems, and work together towards our common goals of understanding, adapting to, and mitigating climate change. Thank you. That's all.
Thank you very much for the detailed synthesis, Dr. Banaay, and also strongly highlighting the key messages that have come out from this focus group discussion. So now, to formally close this event, let us all welcome Academician Gisela Concepcion, a member of the Biological Sciences Division of NAST. Academician Concepcion.
Thank you, Dr. Iniguez. I'd like to congratulate, applaud, and thank our speakers today for your invigorating and enriching scientific presentations. I'd like to thank the organizers of this second of a series of FGDs on microbes and climate change, members of the Biological Sciences Division, and also the Microbiology Association of the Philippines. Many of us are involved in some form of microbiology research. I'd also like to thank our rapporteur and our scientist who gave an excellent synthesis. Well, in my case, I think that my goal resonates with the NAST FDGs, which would always be to see what can be done about the existential problem that we face today. The climate change issue pervades our lives. and the rest of nature. And it is almost a question of our survival and progress that needs to be addressed by the issues related to climate change. And today, that is focused on the microbes. The microbes are invisible. They are small, yet they are big. And so we say small is big. They are powerful. They are intelligent. Likewise, big is small. The ecosystem that we live in, our planet, relies so much on what's happening among its smallest life forms. So this is a philosophical question that needs to be addressed, and no better place to address this than in the National Academy of Science and Technology in the Philippines for problems relating to the Philippines. Having said that, while we need to address Philippine problems, we need to have a local perspective. A local perspective down to the communities, to the regions, to the nation, to the archipelago, but also relating to the Southeast Asian region and the rest of the world. Because for this particular complex problem, we know that the impact pervades us over time and space. And so from the philosophical scientific viewpoint, I would like to bring up the motto of a great learning center of the world, which is Harvard, Veritas, truth. On the other hand, for such a complex problem that changes over time and space, we can only aim for partial truths. Then its neighbor, the other great center of learning of the world, Massachusetts Institute of Technology, has as its motto, mens et manus, mind and hands. And this brings me to the second point, which is the scientific perspective. As we know, science, I think we all believe in this, science is the high country of our minds. But then it takes more than mind to understand our problems, unravel the mysteries of nature. We need our hands. So mens et manus. And with our hands, we have extensions of our senses, which are our instruments, our technologies. And so I was happy to hear something that resonates with what I know of a bit from my own research. I'll just mention them briefly because I'm sure that each person in the audience felt the same way. Something that you've done in your scientific career or you're still pursuing was also mentioned by one of our 4 exceptional speakers, or all of them. So in the case of Professor Leach, I affiliate very much with the microbiome study of plants because I have studied shipworms and their microbial symbiotes. And my group has published on metagenomes and individual genomes in the consortium of bacteria, and also on the metabolomes and the individual compounds that these microbial symbiomes have produced for the benefit of their hosts, the shipworms, which are actually bivalves, but also now being explored as possible therapeutics for human disease. So in the case of our second speaker, Dr. Sabino, I'd like to highlight the importance of the soil microbial amendments that the UP Los Baños is very prominent for, in particular, products of UP Los Baños Biotech are focused on biofertilizers. In particular, I could mention one brand, Microbes, which is a consortium of 15 naturally isolated and combined microfungi that are symbionts of certain plants. And we know that they grow hyphae. And they function to get the nutrients from the soil and also to serve as deterrents to pathogens. In the case of Dr. De Los Reyes, I was waiting for somebody to mention the bio-circular economy. And he did it so well. And in the final analysis, we need to see how we can keep on reusing, recycling the rich nutrients that are found in our natural sources before they escape as greenhouse gases. And I am particularly interested in the water governance initiatives of Dr. De Los Reyes. Finally, in the case of Dr. Dio Onda, well, my colleague at the Marine Science Institute, I think it's very important, Dio, that you mentioned the need for data gathering and analytics over time and space. And let's just say that the data that's gathered must be accurate data. So good data, okay, as a basis for modeling and for forecasting. So I like to say that all the tools, all the strategies of science need to come to play, into play, to address the climate change issue for the sake of our Earth, for our future Earth, which is a one Earth. And we have here national scientist Lourdes Cruz, who leads the Future Earth effort in our country. With that, I wish to thank everyone again for your invaluable contributions to this second of a series of focused group discussions on microbes. And I'd say we just have to bow in awe of nature and know that there are principles that maybe we should uphold, and that is to balance cooperation and competition between the big guys and the small guys. We tend to think of ourselves as the most advanced creatures on this planet. But then, we have to think of plant intelligence, animal intelligence, viral intelligence, bacterial intelligence, and we need to have a modus vivendi with them for us to survive over the long term. The part that I missed at this event was the part on governance, e-governance. And e-governance to me is very important. And this is where the communities that need our help would benefit from if we provided economic governance and ethics governance. So ethics is very important in the way we will provide advice as scientists to these local communities. And I'd like to say that to the 400 or so participants who attended our event today with various backgrounds and various areas of responsibility in all parts of our country, please be vigilant about this problem that we face today. And I would like to think that this scientific session gave you a lot of new learnings. On the other hand, Dr. Onda, our young, outstanding scientist, tells us that they are few and there needs to be more masters and PhD students and graduates who will embrace this complex problem that we face today. And so I would like to suggest that NAST would move forward with implementable actions to create such formal courses so that Dr. Onda, Dr. Sabino, and hopefully Professor Leach and De Los Reyes can continue to teach, mentor, and guide our graduate students to pursue high-quality scientific research. With that, I'd like to end my remarks, and I hope that all of us feel elated about what transpired at our event today. Thank you.
Thank you so much, Academician Concepcion, for providing your own feedback and much food for thought regarding this topic, and also on a larger picture about science and how we move forward. So I think Dr. Raimundo, is there, you had your hand up, or? Okay, sorry. Okay. So with that, before everyone goes, so we would like to invite you as well to the third and last episode of these focus group discussions on microbes and climate change. And that will be held February 17, the same time. So this registration link is now on the screen. So please do participate in this as well. And if you could kindly answer the NAST short survey as well before leaving the Zoom webinar, and reminder that the participants who accomplish the evaluation form, are entitled to receive a certificate of attendance. So that you will be updated about when these events and these discussions, these really informative discussions, will happen again. Please do follow the NAST official Facebook, Twitter, and Instagram accounts. And lastly, on behalf of NAST Philippines, thank you so much for participating in this very interesting and fascinating discussion, and all the efforts from our speakers and all the questions and comments from the participants. So thank you again, and we hope to see you in the next discussion. Thank you, everyone.
Thank you, everyone.
Thank you, everybody.
Recording stopped.
Welcome to the Philippine Science Heritage Center's new Salinlahi Science Center, honoring our top Filipino scientists and their game-changing contributions to the world of science. Know more about all of them and let the Salinlahi Science Center be your gateway into their minds. Salinlahi.